Can you hear me? Okay, awesome. Whoa. Hi, everybody. We're going to start in just 5 minutes, just so the rest of the people can come in. Just letting everybody on the live stream know, too. Thank you for your patience. Hello. Oh, okay. We have to lean in when we talk. Okay. I'm nervous for you guys. Okay. Cool. Okay, I guess you don't have to lean in, just project. Good morning, everyone. Welcome to day 2 of BDC Summit 2024. And welcome to our viewers online who are joining us via live stream. Today is the day. It is the day. I haven't practiced this script. Okay, so it'll be a fast-paced day filled with presentations from our top 8 teams, talks from outstanding speakers, 2 performers, and of course our closing award ceremony where we'll hand out all of the BDC Summit 2024 prizes. We'll be announcing the top 8 teams very shortly, but first we have a few thank yous, as always, that we need to share with you all. So firstly, a very special thank you to the team at MoMA for hosting us for the 6th year. I can't say in a row because the pandemic ruined it, along with many other things. Special thanks to Sean, Carson, Hunter, and the Film and AV teams for all of your support leading up to and during this event. They make everything so easy on us, so we appreciate you guys every year. Thank you to our sponsors for continuing to believe in us, support us, and engage with us year after year. Thank you to Science Sandbox, sponsor of the Science Sandbox Prize for Public Engagement for the 5th year in a row. Thank you to the Siegel Family Endowment, the NYC Cultural Affairs, Department of Cultural Affairs, National Endowment for the Arts, New York State Council on the Arts, Cliff Family Foundation, BASF Foundation, Twist Bioscience, and Tom Tex. We wouldn't be here without you all. And a very special thank you to our judges. Our team appreciates every year the time that you guys spend all the weeks leading up to and these really, really long 2 days. So thank you all for your sustained attention, for your dedication to our students and to us. And that's me.
Cool. So we'd also like to thank our mentors. They're artists, designers, scientists, and so many other backgrounds who took the time out of their schedules to visit the classroom, speak with the students, give feedback, give guidance, and help the teams realize their projects. We had over 30 mentors working in BDC classrooms around the world, and many of whom accepted our invitations without hesitation because they care that much about supporting the students. So for those of you in the audience, maybe watching online, we appreciate your willingness and graciousness to support the students. And last but not least, we want to give thanks to our students and instructors. So this is the 9th year of BDC, and we continue to be awed and inspired by you all. You make the competition as special as it is year after year, and we're so proud of you. So let's have a quick round of applause for everyone joining us here today. All right, so the big moment. Let's get to what everyone's been waiting for, the announcement of the top 8 teams. This is it. We got Apple Braille, American Typewriter. Just more suspense, don't worry.
They're doing this on purpose up there.
There we go.
Wait, what?
Oh.
Okay. Wait, we can advance on here, right? We'll find out. Okay.
So first of all, we've got Maryland Institute College of Art, Symbiotic Stitches.
The second team is Parsons School of Design, Awayste.
All right, third team, College for Creative Studies, Salt Mines.
Team number 4, Universidad del Istmo, Canbe.
Five, we got UT Austin, Consume Our Consumption.
Next is Moholy-Nagy University, pHen.
Seven, we got Rye Country Day School, Polynation.
And finally, McGill University, Kelserra. All right, congratulations to the top 8 teams. We'll give you all some time to shake it off because you're gonna get up here and present your projects again. We'll have a keynote and a performance in between that. But please note that even if you're not part of the top 8, we have so many outstanding prizes that you're still being considered for. So stay here, right? Stay with us. Now let's move on to our first performance of the day. Presented by a very close friend of Biodesign Challenge and supporter. She's been for quite a long time, bioartist and BDC community member Laura Splan. I'm going to do a very quick intro, Laura. Laura Splan's A Guided Sublimation was originally commissioned as a full dome experience by the Vanderbilt Planetarium under the curation of Paul Rubery. The work uses epigenetic science to explore the interconnectedness of the natural world and the built environment by incorporating research on environmental influences on gene expression. The animations combine 3D molecular models with AI-generated landscapes. The accompanying soundscape was created in collaboration with a theoretical biophysicist, a biotech lab instrumentation engineer, and a heavy metal musician. Free bookmarks at the BDC registration table will lead you to more information about this meditative audiovisual experience that invites you to sit back, relax, and open your eyes. So thank you, Laura, for joining us. We're going to hand the stage over to you.
Welcome to this place for gathering together, for gathering bodies.
Our bodies.
Allow me to be your guide. Take a moment to unground yourself, feeling the weightlessness of your body, your limbs, your fingers, your toes.
Your
For
bodies.
this journey, you will need to imagine yourself as a multitude of entangled entities.
Imagine
They are all you. You contain them all.
everything.
Recenter yourself here in this expansiveness, outside of what is known.
Recenter.
How will you reorient yourself in uncharted territory? How will you mark this place so that you can return to it at a later time?
Reorient.
To begin, allow yourself to breathe in deeply, more deeply than you have ever breathed before. You have so much room after all. As you inhale, feel your connection to the space around you. As you exhale, feel your delicate, expansive length, reaching out in every direction, holding secrets of life, not just your life, but the lives of those before you, lined up in succession like pearls on a necklace, passed on from one generation to the next. Your ancestors reaching out as a reassuring tendril to find you in the darkness. Your patinas reflecting each other, memories, dreams, trauma, triumphs, moments. You are filled with immense information. Feel the vast space around you. It's not an empty void, but a dynamic environment. You are the main attraction, chromatin, a complex entity, at once biology, chemistry, physics, ecology, psychology, astrology, ology, all the ologies. Feel the forces of attraction and repulsion between your molecules, the shifts and movements in response to thermal fluctuations, a flexible chain. Sense the viscous nature of your surroundings, a fluid world that simultaneously offers resistance and support. Feel its pressure on your back, your arms, your head. Now shift your awareness inward to the beads on your string, pearls, secrets, a spiral staircase of 146 base pairs of DNA, adenine with thymine,
Together,
guanine with cytosine,
together,
wrapped around 8 histone molecules, 2 copies each of your core histones, H2A, H2B, H3, H4.
your secrets.
These tiny proteins with a high proportion of positively charged amino acids, lysine and arginine, draw in your negatively charged DNA backbone. Electrostatic attraction gently guides the tight wrapping of your DNA around your histone core.
Attraction,
Opposite charges causing affinity between your histones and your DNA.
affinity,
With every inhale, feel the embrace. Like a long hug, 2 meters of DNA wraps tightly around your histone octamers 1.65 times, creating a close bond, forming a protective circle around your supportive proteins. A nucleus of connection, your nucleosome. Divest yourself of the weight of the central dogma. Allow the incompleteness of neo-Darwinian genetics to float around you, suspended with the notion that DNA holds within a single cell all the genetic information needed to create an entire organism. Reimagine what we mean when we say entire organism, permeable membranes, genetic landscapes, a complex gene regulatory network. Your flexibility is a state of mind, sometimes activated, genes on, RNA produced, sometimes repressed, genes off, RNA transcription ceases, functional activity quenched. Visualize your pathways, subtle chemistry transcribing messages, gently nudging on and off, whim and fancy. Sometimes your modifications seep through the generations without changing the underlying DNA nucleotide sequence. You are stealthy. Feel the crisp sensations, enzymes adding, chopping, bits of protein, bits of DNA. Listen to the reverberations of the signals they send to each other, whispering whether your symphony should be sung or silenced, staccato or legato, changing the way it is played without changing the notes. Imagine you breathe in the sweet scent of roses. It reminds you of the sun before there was relentless light. You inhale the sweet scent again, but this time it sends an electric shock racing through your entire body, again and again, inhaling the scent of the roses fatty acid derivatives.
Imagine.
Imagine, after the roses have long gone, the future grows inside you. Weeks later, the future arrives, sitting under a bush, its white fur shining bright under the sun. A floral aroma sends an electric shock through its body, your body.
Our bodies.
Visualize the complex array of enzymes you use to unpack, repack, remodel. Your shape-shifting nature and iridescence of form affects the reading of your genetic information, how your future is seen through a prism of the past. Various stories from the same DNA, anything but an open book. Choose your own adventure. Embrace constant change, multi-protein complexes, moving, restructuring your form, shifting contacts between your histones and DNA, regulating access to your genes, determining what is hidden or heard, defining status, defining fate. Stretch into the flexibility of your unstructured histone regions, intrinsically disordered proteins. Sense the chemical modifications. Feel yourself relax. Your DNA unwrapping, allowing your story to be read. RNA transcribed, allowing your story to be passed on. DNA replicated. Feel sensations of connection as enzymes gently mark your amino acids with molecular modifications, regulating gene expression, expression patterns maintained through cell division. Visualize these marks, some activating genes, some repressing genes, some able to do either, depending on where the mark is placed. Location, location, location. Imagine it's the early hours of the morning, a mist in shrouds, rows upon rows cropping up, delicate, their petals not yet exposed, protected from nature, from you, from itself at all costs. Imagine cutting each stem as instructed, noticing pointed thorns, your calluses immune to their piercing touch, noticing fleshy petals, your calluses immune to their velvety texture. Imagine your focus turning to fuzziness, exponential, blurred vision and dizziness set in. It is just the onset of a series of recent events, iterating themselves as fractals in the present. Take a moment to notice the marginalia of your own compendium. Protagonists, antagonists, proteins, enzymes. You give them nicknames to help you remember who you are. Readers bind to you with a warm embrace. Writers carefully place marks on you with a tentative tingle. Erasers gently remove marks from you, working simultaneously, everything all at once. Reading, writing, erasing, activating, repressing, over and over. Your story is constantly revised, edited, a living document. Transcription errors, misattributions are to be expected. Accept that you are not always understood. Some say your epigenetic shapeshifting is a kind of code that highlights and obscures your genetic code, shimmering zeros and ones, recursive loops and functions, strobing on and off, a semaphore message on the distant horizon signaling the future. an encrypted file decoded on a subliminal level. As you compress and decompress, passing moments into the darkness of now. But your modifications are not consistent, predictive, and combinatorial. You are not code. Free yourself of such semantics. You are otherwise. You are other ways. Imagine you are trying to remember who you are, what you look like. How would you describe yourself? Keywords optimized for discovery, prompts optimized for results.
Engineered.
Imagine you are no longer able to conjure an image on your own. Perhaps you want to unlock more possibilities for how you can be conceived, for what you can conceive. So many styles to choose from, so many parameters to enable.
Chaos.
Imagine yourself. Imagine yourself imagining yourself.
Imagining yourself.
Explore the possibilities of your expansive repertoire of histone modifications. You are not merely winding aimlessly. You are following a choreographed path, orchestrated by chemical kinetic constants between the negatively charged phosphate backbone of your DNA and the positively charged regions of your histone proteins. It's a dance of electrostatic forces. Feel this charge, this push and pull, the energy of chemical attractions. You are not static.
You are dynamic.
As you continue to breathe, feel the freedom in your form, the rhythm as you sway with the gentle flow of your nucleoplasm. Consider the paths you could follow, or will they follow you? In this expanse, you can feel the presence of enzymes, roaming, scouting for specific cues, modifying your histones, adding and removing chemical groups, altering your form,
Relaxation, compaction.
At times, you feel the touch of acetyltransferases, adding acetyl groups to your histones, making you more open,
More relaxed.
but now you sense the tug of deacetylases, pulling those groups away,
Coaxing you to condense.
You are becoming increasingly aware of your need to compact, increasingly aware of your intention. You are protecting vital genetic information. You are information to be stored, mapped, plotted in infinite ways. You like to keep them guessing, suggesting legibility, logic, order.
Resisting interpretation.
Visualize yourself as data visualized, exploring axes,
X, Y, Z.
locations scattered, time stretched, stories encoded and decoded.
Looking back, looking forth.
Sometimes you catch a glimpse of your own reflection, and you whisper to yourselves, if it were up to us, our eyes would merge, suspended, before the moment shattered into the kaleidoscope of later. Now, as you inhale deeply, start to feel yourself drawing inwards to a tighter, more introspective state, a succession of nucleosomes linked by short stretches of DNA, about 20 to 60 base pairs in length. Your closest sibling is 25 nucleotides away, or 0.0000000085 meters. You feel yourself attracted to them through the mediated interactions of a transcription factor, pulling you, pulling them. Feel the maximum compaction. Take solace in possibilities for redemption. Your regulatory processes are dynamic and reversible, on and off and on. You are forgiving. Repressive marks can be removed. Active marks can be added. You are kind. Allowing cells to select from the complete genome only genes needed for a particular function, which can change with time. You are resourceful. You have covered a lot of territory. You think you won't be able to remember it all, but remember, you are not alone. You contain a multitude of entangled secrets, unlocked by all your senses. You will remember how it felt. It will be a gut feeling that you will no longer ignore.
Thank you, Laura, for that wonderful performance. I usually meditate every morning, so I was glad I was able to get that in today too. So now moving along to today's keynote speakers, Marco Poletto and Claudia Pasquero of EcoLogic Studio. They will be joining us via Zoom. I'll read a quick introduction of EcoLogic Studio, and then we'll begin the talk. So EcoLogic Studio is an architecture and design innovation firm specialized in biotechnology for the built environment, co-founded in London in 2005 by Claudia Pasquero and Marco Poletto. The studio has built an international reputation for its innovative projects integrating systemic thinking, computational design, biotechnology, and digital prototyping. Their new book, Biodesign in the Age of Artificial Intelligence, Deep Green, investigates the potential of nature-based technology for shaping the evolution of contemporary architecture and design. It takes on the now pervasive topic of design intelligence, extending its definition to encompass both biological and digital realms. We're very excited to have Claudia and Marco joining us virtually for this talk. Thank you so much, and take it away.
Thank you very much for the wonderful introduction, for having us. I hope you can hear us fine. And I will try to share the screen with sound. There we go. If it's possible, and you can let us know if you can see the screen. Now I will try to.
We're currently seeing the PowerPoint application, but not
Yes.
the slides.
Which looks slightly stuck at the moment, but I think we should be getting there.
There we go. Looks good on our end.
Excellent, excellent.
Thank you.
So, I mean, you already introduced us, so thank you very much for that. I'm Marco Poletto, And this is Claudia Pasquero here with me. And also, you kindly introduced our new book, which is edited by Routledge and has been around for a few months now, and essentially really collecting the last 10 years of project as well as design, innovation, and research that we have conducted. And investigate the potential of nature-based technology for shaping the evolution of contemporary architecture and design. And we look at the topic of intelligence, and of course, these days there is a lot of emphasis in artificial intelligence as a kind of a paradigm of computation and computational. Believe that biology and biological systems have a lot to give in terms of what it means to be intelligent today, and in which way the technologies such as AI may become instrumental in evolving our relationship with the living world and with the natural biosphere, rather than being detrimental to it. So our work really tried to investigate that, and in this sense, we operate at different levels from by digital design with work that is often as kind of artistic or sculptural applications to the issues of urban wellness and well-being. So how architecture creates spaces that are restorative of our body and minds, as well as the issue of food and urban agriculture. So how to integrate biodesign innovation in our cities, making urban environment. And of course, are you saying something? Can you hear us okay? Good. And of course, the topic of carbon neutrality. How does it becomes generative in terms of a new form of architecture? And I mean, if we are serious about carbon neutrality or the future of our civilization, we need to look at what to do with what we call the urban sphere, which is essentially this network of infrastructure that supports us, supports our urbanizing civilization, and it will need to support us for many years to come, because despite a kind of slowing down in the rate of growth of a global population, still we will have more than 6 billion people living in cities in the next 20, 30 years. So it's a huge amount of people that will need to be supported and fed essentially by infrastructures that at the moment are distributed around the planet, and effectively creating what is a linear and open metabolism. And that's where the problem starts to emerge. Open metabolism means that cities need resources which are often extracted from the biosphere still today, and generates a huge amount of waste or pollutants that are still ending up in the biosphere in many respects. So still today, despite all the progress, we are relying on the natural systems of the biosphere to recirculate these waste into new resources. And so, of course, the biodesigner faces a big challenge if we want to be serious about the role of biodesign architecture in our future. And it is really to find ways and solutions to essentially convert what cities expel in the form of waste or pollution into a resource, into something that we can use to sustain our civilization and build the spaces that host us. So a few years ago, when we started the Photosynthetica project, we kind of came with the very basic diagram whereby the Photosynthetica design system would need to be able to do that, to essentially take in inputs and then generate useful outputs which would then be recirculated within a circular metabolism. And why algae became one of our most powerful allies in this process is really because in the biosphere, in the living world, they are already the organisms that potentially do the hardest work. Carbon and recirculating this resource into biomass. And this, they do it in the oceans, they do it in water bodies, and they have been doing it, if you want, since the beginning of our biosphere as we know it. They were, in fact, the first organisms that contributed to the creation of the atmosphere that we breathe. And so essentially, they and for in biodesign bring their abilities, their intelligence within the built environment as powerful engines of architecture. And if we look at it at a quite basic level, first observation when we compare them to plants or large trees is that microalgae are 10 times more efficient in photosynthesis because their entire body and their entire metabolism is dedicated to that. Whereas plants, of course, as complex systems, have other requirements to support them, which means that integrating them in architecture gives a much higher potential to transform buildings into photosynthetic organisms themselves capable of remetabolizing pollutants and transforming it into useful resources. Of course, the sun is the driving force of photosynthesis, and the carbon dioxide and pollutants that are present in the urban atmosphere become nutrients for the algae organisms that are able to feed on them and grow, duplicate, create biomass, nutrients, and this circular process was subject of a research development into biomaterials and circular metabolisms, which then was essentially transformed into a productive space last year in the center of London called Air Lab, where we actually set up a workshop, biodesign workshop, that was accessible to all in the public. Living organisms could effectively capture pollutants from the atmosphere of London and turn it into biomass, which we could then convert into biopolymer and then 3D printing filaments kind, in this case, an epipod, which is a product of self-care, demonstrating in a way how by taking care of the planet, we can also take care of ourselves. So in a sense, design becomes quite literally between us, our society, and the living world that surrounds us. This project was also awarded kind of an award from Fast Company, and the origin of a series of projects that we have completed in the last 12 months, bringing this type of benefits to the workplace, to the work environment. This is the Air Office. It's a restorative space we built in the headquarters of Helion, which is a healthcare company based in Switzerland, in Nyon. And it kind of demonstrates again how algae within any urban environment, and in this case, really in the space that we occupy most of the time during the day, our work environments, bringing purification, but also bringing a soothing experience of connection with nature. And so the idea is really through this type of design innovation to introduce systems where these kind of microorganisms are allowed to become our companion, our symbiotic systems in kind of breathing, in transforming the atmosphere and the spaces we inhabit. Here are some photos from the project. It's been run and it's been received very well also from the employees of the company. In fact, initially it was supposed to be more of a test project, but it has very much now become a permanent feature of their workplace, and new ones are in progress as we speak. And here are some of the product that we have produced for Helion using the algae biomass, the algae biopolymers that we extract from the purification process itself. We also embarked into product design development of Air Office, which is called Air Reactor, which aims at creating a scalable solution of this innovation. We call it the first desktop biotechnological air purifier. And essentially it is exactly that. It's a desktop version of the Air Office, and it allows people to grow their own personalized culture of algae in the office as well, domestic spaces, and to essentially bring this solution pretty much into any urban environment. As here it's a chart describing the amount of input and output that this the air reactor is able to create, and how do we effectively transform pollutants into nutrients that feed us. From the design point of view, also, you may have noticed from the photos that we are really pursuing actively an idea of using digital fabrication and prototyping to create products that are entirely reversible. So there is no use of glues and other type of artificial binding agent. All components are just simply assembled through frictions and connecting pieces, and they can be disassembled, reused, recycled, and composted for the biopolymeric parts without any waste being produced. And this is the diagram that describes exactly how that can work. And of course, this is Lulu, which is my daughter, and she's testing the air reactor, showing how you can harvest the algae from the reactor. So after the cultures have grown enough, it is possible to extract them in a very simple way. I mean, the whole system is really designed to be intuitive and user-friendly, and allows essentially anyone to harvest them and make use of the algae, while the culture is then allowed to grow further and until the next harvest time is ready. So they keep replicating, they keep going, and they can be kept alive for a long time. The same system was also implemented in the headquarters of Nestlé in Portugal for a collaboration that was looking more at the food aspect of microalgae. Some of you may know algae like spirulina or chlorella, which possess a really wide palette of properties in terms of being nutritious and being being able to supplement our bodies. And essentially, this system again demonstrates that it is possible to grow them within the environment. And in this particular case, it was part of the headquarters space, and they had the sort of employee kitchen area on one side, but also very much visible on the other side as a part of the main entrance of the building. So again, it's not only a structure that brings new opportunities, but also brings a new language, a new understanding of how architecture becomes alive and contributes to the future metabolic properties of our cities. This is maybe like a second chapter of our presentation today, and it's kind of looking at some of the opportunities that this type of approach is generating at the scale, at the larger scale, if you want. And of course, we use different opportunity to test these logics, these ideas, and the project 3.1 was actually created in collaboration with Hyundai Motor Company in Korea, in Busan, and then Seoul. And it kind of starts from their vision for how to express a new society that can live as a carbon neutral society. And we tried to interpret that through first a piece, like an artwork that you see here, which is called 3.1, and that demonstrates how the technology that is currently used in the fabrication of cars can be repurposed to build photosynthetic and carbon neutral architectures. So this tree is looking at the archetype of a column and kind of reimagines it as a living organism, and it hosts living cultures on its trunk, but it's also itself made of the biomass grown on the tree itself. So it becomes a storage of carbon itself. And there's a little video that describes the ideas behind the project.
One of the most significant aspects of contemporary technological evolution is its inevitable convergence. 3.1 celebrates this epochal transition. On the one hand, it is a living tree, our most potent symbol of nature. On the other, it embodies a new kind of technological life, designed by artificial intelligence and biodigitally grown. 3.1 has the same photosynthetic and carbon capturing potential of a small forest. It metabolizes and stores the carbon molecules into its 3D printed bioplastic structure while releasing oxygen in the atmosphere. Its main structure is a 3D voxel grid developed by a trained AI algorithm whose recognition of arboreal systems negotiates the logic of the architectural column, the new archetype of Habitat 1. The trunk and canopy are robotically 3D printed with algae-based biopolymers, derived from the harvest of the same living photosynthetic microalgae cultures that grow within its 42 glass reactors. The mechanical strength of its biodegradable structure is derived entirely from its unique pleated morphology, inspired by the fibrous trunks of actual trees. The production of 3.1 as a carbon-neutral and zero-waste structure required the setting up of a global informational system capable of running a remote fabrication process involving 4 industrial robots and 20 large-scale 3D printing machines. Moreover, the custom software and hardware was developed to adjust the morphology and the printing process in order to eliminate the need for support material, as well as to accommodate the material properties of algae-based biodegradable polymers. This process demonstrates how digital and automotive technology can be used to manufacture the bioarchitectural components. 3.1 is the testimony of the dawn of a new technological era where the contemporary digital and robotic infrastructure is repurposed to fabricate or grow soft, wet, living biodigital products. The age of Habitat 1.
So we are often asked whether it's possible to grow architecture, no? Or whether at some point in the future we will grow our cities and our buildings. And of course, it's an answer that is difficult to give, but I think this project was our attempt to show how close, or what is one way to embark into this challenge. The challenge to think about architecture as a grown living system rather than as a built assemblage of technological parts. And so in that sense, it was a very inspiring journey. And we just of speculative videos and images using AI algorithms that were somehow helping imagine or project what a technological system like 3.1 could mean or could do when seen at the scale of the city itself. And of course, this was then exhibited in Seoul, and the real 3.1 was installed within the Hyundai Motor Studio space, but also on the facade of the building, we kind of projected this vision of a city that is essentially grown through biotechnology and design. These are some of the images of the same space from the inside, with again another video projecting it into the city. Perhaps in this journey of dreaming of an architecture that can be grown, this project Ortus Excel was one of the most significant. Probably is one of the projects that have been published and exhibited more from our work around the world, and certainly one project that it is very dear to us. It was originally commissioned by the Centre Pompidou in Paris. This is the photo of its first implementation there, with the two of us, kind of taking care of the living cultures of cyanobacteria within it. And in this project, we tried to really push the idea of a 3D printing as a technology that allows us to design at the scale or at the resolution of the microbiome. In other words, we can create architectural components or structures that are very finely articulated in three-dimensional space, so that they become receptive to biological life. Like in this case, the structure, the sculpture of Hortus really is just a scaffolding for the colony of cyanobacteria to grow within it. And in this case, since the matrix is open, is porous, the colonies are not growing in water, but they are growing on a biogel, on a kind of jellified solution, which keeps the shape while at the same time retaining the necessary humidity and water content to keep the cultures alive. Of course, the sculpture requires a quite large amount of maintenance, in a sense that it needs to be kept humid and wet, which is a characteristics of essentially all living system. And the same as we did for 3.1, this project also became a kind of speculation to project these type of processes at the scale of an architecture, of a large tower in this case, which would operate in similar way as a filter of the urban atmosphere, really mining for nutrients from the atmosphere and converting them into biomass that can then be utilized within the thickness of the space itself. So from the inside, it becomes a very much a kind of large vertical laboratories or biotechnological space where different type of programs evolve around the availability of resources. And one of these programs, we were able to actually build it in one-to-one scale as a kind of unit, or actually as a three cells, a three unit of this kind of a photosynthetic tower building. This was actually in occasion of the expo in Astana, where we were able to create 3 cells of photosynthetic architecture, each one of them representing or embodying a different way of engaging with life around us. So through science, of course, the space of the lab of rationality, where we use the tools of science to learn and discover nature and its unique properties, but also through the space of art and experience, the living space, where we essentially immerse ourselves in the patterns of life, and we experience this type of connection through a form of language that is more meta, if we want. It's more operates through intuition rather than rational understanding. And then finally, we created the third space, that was the space of craft and production. So it's the third way, let's say, to connect with nature, the garden. So gardening is a way of essentially cultivating or culturalizing these type of organisms within the urban realm. So the project, of course, at the same time was also a demonstration, a living demonstration that it's possible to build essentially living algae farms at this scale. And it had quite a lot of technological innovation within it. Was able to host around 1,600 liters of living cultures within these coils of glass, borosilicate glass tubes. The final chapter of the first part of the book, which is also in a way the third chapter of our presentation today, really connects to the bigger issue of reprogramming the blue-green city. So beyond the architecture scale, we needed to engage with topics that are of planetary significance. And a few years ago, 2 years ago, we were invited to take part to the COP26 conference of global climate change in Glasgow, and we brought this pneumatic system, this pneumatic architecture called Air Bubble Eco Machine, to demonstrate how a carbon neutral architecture should feel or look like if we really take seriously the notion of carbon neutrality. Essentially, we need to think about an architecture that is made 99% of air, water, and living organism, and where the fine membrane, the fine line that separates the interior from the exterior, it's really just nothing more than the thinnest of skins. And in this case, it was very much inspired by the biology, but also the mechanics of the cell. And it was hosting living cultures into this membrane of TPU. The project also traveled to Saudi Arabia, to Egypt for future conferences, and became a little bit of a manifesto for the idea of an architecture that, even if small in scale, has potentially a large significance in terms of the impact it generates. And the same can be said for Air Bubble Playground, which is probably one of the projects that is more that has received more praise for what it has tried to achieve, and essentially tackling the issue of quality in the urban realm. There is a short video that introduces the message of the project. The video was created by Sashi and Sashi as part of the sensibilization campaign of this project.
I feel stuck in the middle. Do I send them out to play in collusion, or keep them locked up inside.
Is a major problem. Pollution levels are far worse at the child's height. They are more sensitive to the detrimental effects of air pollution.
We all grew up going outside and not having to worry about things.
I never thought I'd have to protect my kids from the one thing that keeps them alive.
As a mom, it breaks my heart.
I just want to play.
Microalgae are incredible organisms. And with our technology, we can use them to clean our city's air. We have gone to one of Europe's most polluted areas and built the world's first air-purifying biotechnological playground.
The uniqueness of this concept relies on the relationship between the architecture and the kids.
Have fun there.
The more children play, the more the algae absorb pollutants and clean oxygen is released.
This algae is what's taking the bad stuff out of the air.
The air feels amazing.
The key aspects of this project is to create awareness that it is indeed possible to clean the air we breathe.
This can't be the only way to help them breathe better. We need to do more.
So the project, again, really demonstrates that it is possible to break down big challenges, like, for example, the one of air quality in cities. And through biodesign, we can tackle them right here, right now. Essentially, we can begin to create solutions that even if they operate at a small scale, they can generate immediate relief, immediate options that are beneficial to anyone living in cities. And then through the application and the scaling of these approaches, potentially have much larger impact, both directly and indirectly through the, let's say, awareness that this project raises, especially the possibility to experience directly the benefit of biotechnology in urban design. So really the project is one of our ongoing commitment to show that biodesign has effectively the possibility to be solving problems, creating exciting spaces, and essentially reconnect us with the natural world in the immediate and without waiting 30, 40 or 50 years before something is happening or can be done. From the technological point of view, again, the sort of active element is the green ring of glass reactors. That's where the algae cultures live. The air, the polluted air is inoculated from the bottom. It raises through the liquid medium when it's purified, and then the clean air and oxygen is released from the top within the bubble. The bubble is maintained by a thin membrane of ETFE that creates a microclimate of pure air inside. The project had a series of sensors embedded in it, which we used to monitor the efficacy and effectiveness of the system, and we had pretty encouraging results, up to 80% almost reduction in pHen 2.5, which is fine particulate matter, and also near the same percentages for the nitrogen dioxide. So essentially, really the possibility to get immediate benefit is there if we dare applying it. And again, luckily the project also had received a lot of success, and we are really now in these next few days on site completing a new version of this, also in Neon as a garden space, as a restorative space. We call it biotechnological healthcare garden, It integrates not only microalgae, but also medicinal plants all around the pavilion itself and the beautiful sitting area within it, which is accessible to all employees and visitors of the Helium campus. And again, demonstrating how we can effectively bring biotechnology in the built environment today and also engage in beautiful new type of restorative environments. So to conclude today, because I think we're running out of time, of course, the shift in paradigm here is to think about architecture no longer as, let's say, the kind of modernist machine for living, like as a container of programs and functions, but more as a living machine, essentially itself becoming a dynamic process of production. And, you know, from this point of view, of course, also the city itself acquires a new meaning, a new significance. And we are now, since a few years already, exploring the possibility of looking at organisms such as algae, but also slime mold and myceliums and others, not just as materials for architecture, but also as intelligent forms that can help us plan and design better spaces, better architectures, and better cities.
In this case, we have developed a project for the Centre Pompidou in Paris, which is now part of the collection. The Gamphizarium deploy the intelligence of green plants in Paris. So we train an AI algorithm
slowly simulates the future of Paris, where the results of the green infrastructures of the city are evolving and co-evolving with the fabric of the city. It is a different scale, super very large scale, when Paris is seen only as a kind of a rhythm, and slowly but it's a video where we think about speculation more as a simulation of possible futures, and especially
Hello? All right, thank you, Marco and Claudia, for the presentation. We're gonna move into the student presentations now. And sorry about all of the audio glitches and stuff we were having, but yeah, we'll see you next time, and thanks for joining us.
Thank you, guys. I hope you managed to hear and to follow. But yeah, it was a pleasure. And thank you for having us once again. And good luck for the rest of your evening.
Thank you. Have a good day. All right, so we're gonna shift now into the top 8 student presentations. Wow, that's so cool. It's me. So we're gonna start in the order that we announced the projects. So the first team that's gonna be up is MICA with Symbiotic Stitches. So just so you all know, we have a clicker up here, and once you're up here, I can show you all how it works. We also have these microphones here. So if you all wanted to kind of spread out a little bit on stage, feel free to grab one of the microphones. And so it'll be the same experience as yesterday. We'll do 15 minutes for presentation, 10 minutes for the presentation, 5 minutes for Q&A. We'll be giving you time signals, and then we'll be running the Q&A with the judges from the audience. Wherever you want, you can use these microphones. This will advance a slide, and this will go back.
Hello?
So if you want to use the handheld microphones, just make sure they're switched on.
Switch it? Oh.
Hello, we are the Maryland Institute College of Art Grow the Futures class. We collaborated with 3 organisms, mycelium, lichen, and slime mold, to create a bioquilt. We are Symbiotic Stitches. We began our time together by asking ourselves this question: how can we practice care with our collaborators and with each other to craft resilient, symbiotic communities? Our conversations always came back to care and community. Resiliency is the ability to recover and endure difficulties. Care is resiliency. We practice care with our collaborators, mycelium, slime mold, and lichen, by supporting them and not extracting from them. We focused on elements of regeneration to repair and mend in our quest for care as resiliency in our community.
Having been president of the MICA Quilt Group for the last 3 years, I've had the privilege of learning the significance of what it means to make within a resilient community. The MICA Quilt Group is a multi-generational raffle quilt group, and the club has been a place where people across MICA's campus, students, faculty, staff, and even those in the greater Baltimore community can come together. We work collaboratively to create quilts that we raffle off for student scholarships, and we work with many traditional quilting techniques and methods alongside contemporary and experimental ways of making. A quilt is defined and historically understood as a multi-layered textile. Quilts are a significant part of our material, cultural, and creative history, an archive of memory, storytelling, and community gathering. Symbiotic Stitches is inspired by the legacy of the MICA Quilt Group. Bringing biodesign and material experimentation into an intersection of craft and science will make these spaces more accessible for the future of our relationships with materials, where biomaterials can become an intergenerational exchange grounded in familial knowledge and care. Our quilt is made up of various quilt squares that were grown, patterned, and dyed with 3 different organisms: mycelium, slime mold, and lichen. With these 3 organisms, we have spent many months understanding the symbiotic relationships between them and how they work with or even against each other through their materiality. We will now explain the process of how we are crafting the layers of our own bioquilt.
Our first quilt patch is mycelium. Mycelium, what is it? Mycelium is typically noted as the root structure of mushrooms and is part of the wonderful kingdom known as fungi. Not only is mycelium a force within the biodesign materials space, but mycelium grows into large and abundant networks, and we are inspired by this widespread connectivity as an ethos in our project. We were also interested in working with self-regenerative organisms, meaning able to initiate self-repair once exposed to damage. And during our research, we came across an article called Fungal Engineered Living Materials by Elise Elsacker, Meng Zhang, and Martin Dade Robertson, in which they studied the self-repair mechanism of mycelium materials. From this, we wanted to collaborate with mycelium to make self-repair patches for our quilt, in which our quilt could undergo damage but be regenerative. To grow mycelium, we chose our main mycelial collaborator, known as Ganoderma lucidum, also known as red reishi, and we made a mother plate. From this mother plate, we inoculated malt extract broth, which is a liquid nutrient source, and that incubated for about 5 days until we got enough growth. After that growth, we blended a ratio of mycelium growth, more malt extract broth, and 20% glycerol treatment to plasticize the samples, which gave us a mycelium slurry. That mycelium slurry was transferred to several different containers, and most notably a 6-well plate, in which it grew for about 10 days. We added about different fiber samples to grow with our mycelium. We chose wool, silk, and cotton to, one, act as a reinforcement, and also to possibly act as a scaffold for self-repair to occur.
So moving on from our samples, we began to grow our actual quilt patches. So we began to inoculate more Erlenmeyer flasks, and then we moved on from the 6-well plates to Pyrex Tupperware containers, and then we incubated those containers for around 10 days before we took those out to actually press into the samples for 72 hours. We then decided to scale up even larger, and so we inoculated a bioreactor with more mycelium from our mother plate and more malt extract broth, and we let this incubate for 6 days before we took that out to press.
To get our samples, we pressed them. We pressed all of our samples with hemp fabric because our samples grew a bit thin, and it also acted as a reinforcement to our samples. We purposely did not heat press our samples because we wanted to keep the spores viable but dormant to initiate self-repair experiments.
And so from our research, we've determined that mycelium is unable to recognize site-specific damage and rather just grows an additional layer of biomass. And so something that we're interested in seeking out in our research is how we can actually guide that site-specific growth for the mycelium. And so we're approaching this regeneration through the craft of care, which means that we're trying to support our collaborators and not just extract from them.
Self-regeneration of mycelium is vital to our larger notion of the quilt. A quilt mends from damage. Self-repair is not about replacing what once was, but accepting the beauty of change. Self-repair represents a philosophy of evolving from ripped stitches, where we live in a world where materials and generations can mend from within.
We collaborated with lichen to make orange and purple hued dyes for our quilt. Lichen is an organism that is a symbiotic relationship between cyanobacteria and fungi, commonly Ascomycetes, also known as cup fungi. Lichen is a slow-growing organism reflective of the environment around it. The coming together of both the cyanobacteria and the fungi represent this process of lichenization. And we embodied this notion of lichenization in our quilt by collaborating with other organisms, introducing them together, fostering environments for their growth, and therefore introducing ourselves into that relationship.
Lichen contain acids that make it possible to extract dye from. We fermented orco and umbilicaria in an ammonia solution for a few months to achieve purple hues. We started to test the dye on fabric as early as 5 weeks. After our dyes were able to successfully bond to the fabric, we proceeded to dye our mycelium patches. Despite its hydrophobic qualities, mycelium was able to pick up the color. There's another method to extract dye from lichen, and it's through boiling them in water. For our orange hues, we boiled tree lungwort. We were able to achieve a few different shades through pH shifting. We are appreciative of lichen's generosity of color.
We use slime mold for our patterning. Slime mold is a unicellular but multinucleated organism that belongs to the protozoa kingdom. And slime mold is great because it's able to solve shortest path problems by detecting nutrient sources. And so we wanted to utilize this ability to conduct site-specific growth for our mycelium patches. To do this, we needed to find out whether or not these two organisms actually got along together. And so we did some experiments by placing mycelium and slime mold next to each other in the same petri dishes.
Based on our observations, we found they didn't interfere with each other that much until there was not enough food resources for slime mold and it started to feed on mycelium. So we categorized their symbiotic relationship as one of commensalism. What is commensalism? It is an association between two organisms in which one benefits and the other neither benefits nor is harmed. From that, we shifted our attention towards collaborating with slime mold to create resist patterns. Now that we have introduced our 3 patches for the quilt, which are lichen, mycelium, and slime mold, we start trying to combine each two together to foster a symbiotic relationship. Since we observed that the slime mold and mycelium have a relationship of commensalism, they can collaborate with each other to create patterns.
Since slime mold is able to transport substances, based on our research, we learned that if slime mold eats the oat with the pigments, it acquires the color of these pigments. So we tried dyeing slime mold with lichen. We dipped the oat into the lichen dye and placed the oat near the slime mold. The slime mold approached the color oat and acquired this pigment, as you can see in the close-up image here. We also soaked our mycelium samples in lichen dye. The one over here is Oracle, and this is lungwort.
One of the initial ways that we brought all three organisms together in our experiments was through cyanotyping. Cyanotype is a developing process involving UV light energy that transforms materials into a Prussian blue color. To do so, we applied the cyanotype chemistry on the surface of the mycelium. We then exposed the chemistry, turning the surface of the mycelium blue. Next, the slime mold was applied onto the blue mycelium surface, and as the slime mold moved around, it left trails of white. We use cyanotype as a way of documentation between the relationship of slime mold and mycelium.
Here's a close-up of the surface of the mycelium. You can see faint trails of white where the slime mold has consumed the cyanotype chemistry. Building on our notion of care, we work with our organisms and not from. They are collaborators and not just objects to be extracted from. While cyanotyping, the mycelium started feeding and growing over the blue chemistry, creating new patterns of blue and white. Our cyanotypes inspired us to try anthotypes, to swap cyanotype chemistry out for lichen dyes. For the anthotype, exposed areas to the sun fade in color, leaving behind darker impressions of what was above.
Our values are based in the understanding that craft and biodesign stemmed from the need and fear to express and push through times of struggle. With biodesign, we rest with the heavy notion that we as a society must take control and responsibility for the problem that we have started and magnified. The slime mold became resilient in its pathways and growth. It found a way to thrive either way. The mycelium regenerated and healed its holes. The lichen created a warm vibrancy through its hues. It's within our familial heritage to create, to quilt. Biomaterials have a place in that space, making them accessible to the world through quilting, a symbol and process of community. Quilting is a way of craft that has been used throughout history to bring care to resilient communities. We look back to those quilters and move forward with our heavy hearts and minds as we center this craft in our fight towards building resiliency amongst ecological crises.
Here's our final quilt. Here's some close-up images. Thank you to our human collaborators who couldn't be with us today, Jingwen Zhang, Lei Guo, and Austin Chia, as well as a special thanks to Ryan Hoover, Rachel Rusk, and Caitlin Grace McCollum.
And thank you to our more than human collaborators, Ganoderma lucidum, Orchel, lungwort, umbilicaria lichen, as well as Phycerum polycephalum.
Here are our sources. Mine works.
Is this working? No?
Here, you can.
All right, so we're going to take questions from the judges. Anyone have a first question? Oh, all right, Laura.
That was a great presentation. I love this project and how you covered the kind of history of quilting and connected it to the raffle project. And I was wondering if you could talk a little bit more about the construction of this quilt and who was involved and how, if you've had any ideas about connecting it back to something like the raffle project.
Yeah, so basically our group here in this class, we helped craft this quilt, but I've been the president of the MICA quilt group, and so something that I've been kind of interested in is how we're bringing these conversations around craft and care into the biodesign community, and also vice versa, bringing these understandings of what it means to work with organisms back into the MICA craft and quilt group. We work with so many people around the community that have never had access to the biodesign space, and so I think us working together to learn what it means to make a quilt with these organisms can help us to future make quilts with organisms and other types of materials.
Thanks for a great presentation. And your quilt is actually quite beautiful and in its own right a work of art. This is a technical question, but do you know anything about the light fastness or the resiliency of these dyes and these materials?
They are not light fast, but we tried to use that to our advantage, and that's why we tried anthotypes, where we ended up creating some resist patterns.
All right, additional questions from our judges? Yeah, Todd.
Sort of building on that, my understanding of quilts and the ones in our family too, they're sort of long-term, right, that they're handed down. Can you talk a little bit about how the sort of stability of your materials and how they're going to sort of last over time?
Yeah, great question. We are really interested in how our quilt can evolve over time. So a part of our notion and perspective of the quilt is that this will be handed down through generations, but also be able to regenerate. So it'll change once put under different conditions of the different generations that will be able to handle and interact with the quilt.
Beautiful project. My question is about your choice of stitch work and applique as opposed to maybe more traditional quilt piecing techniques. What drove the choice to do running stitch as opposed to others, or in combination with other stitches?
I think the choice for the running stitch was so that we could actually showcase the ways that we're actually bringing together all these different pieces, as well as also just for the stability of the mycelium, as it is quite fragile right now. But thinking about the different layers, I think we want to really communicate that these are all stitched together and that we all were working in from a patch method and bringing all those patches together.
All right, one last question from the judges. Oh.
Thanks, it's a really lovely project. I'm wondering, did you experiment with mordants or any kind of traditional dyeing techniques that enhance the color fastness?
Lichens are traditionally not used with mordants, and that's why we wanted to try them out, but we were changing the pH a bit to try and get different hues.
All right, thank you all so much for your presentation and answering the questions.
Thank you.
All right, next up, could we have the Parsons Awayste team come to the stage?
Okay. Bueno, first of all, I'm sorry, but if any of you is also allergic to AC, I hope you know the struggle. So let's begin. Okay, everyone. So we are Ermac and Lucia, and we're here to present our project Awayste. And we're gonna begin with one slight confession. We are both obsessed with waste. But more specifically, we are both obsessed with packaging waste. You see, product packaging is just a mere means to an end. It's not even the intended product itself, yet it accounts for about 28% of the municipal solid waste that we produced in the United States. Now, as part of our Master's in Strategic Design and Management at Parsons School of Design, Ermac and I have spent the past 2 years trying to understand the waste crisis from a systemic perspective. As part of our research, we decided that we wanted to focus on biomaterials because they offered us a glimmer of hope. We were very excited about several innovations that I'm sure many of you here are familiar with. Your cassava bags, your seaweed bags, bioplastic cups, you name it. All innovations that offered us a glimmer of hope. Now, the Environmental Protection Agency has laid out the following hierarchy to help us understand what are the most preferred actions to tackle the waste crisis. As you can see here, waste prevention is the most preferred strategy. It's oftentimes also referred to as a reduction, so prevention. And then it's followed by things such as reusing and recycling. Now, in light of this hierarchy, we became particularly interested in one specific subset of biomaterials that were being used in the packaging industry that have these particular attributes of being no waste. So from now on, we're gonna call them no waste biomaterials. Now, we brought one specific example to give a much more clear sense of the idea. This is one edible packaging sachet that was launched by UK startup Splan. Now, the idea is that these gels can be eaten by athletes, and so by incorporating the packaging within the product experience, the idea is that ultimately you are eliminating the need for packaging and contributing to waste prevention. Now, the good news is this is not an isolated example. There are multiple examples of no-waste biomaterials across different categories, in the food space, the beauty, and the cleaning industry. Now, we were super excited about this, so much so that we decided that for our thesis project, we wanted to develop our own no-waste biomaterial. We actually decided that we wanted to focus for the fashion industry, and we designed our own bio-based poly bag that was meant to dissolve in the washing machine, serving as a laundry detergent. We called it Made to Fade. It was a great experience, and we learned so much through that process. So actually, when we were able to create our own prototype, our idea was that we needed to start to look for funding. And so we thought to ourselves, okay, if our product is actually successful, we would actually be helping in waste prevention. You know, the government is actually the one that pays for all the waste management costs. So we assumed that it would be not that crazy to think that we could find some grants in the government side that could help us, let's say, fund our project. However, a very quick reality check showed us otherwise. So this is an example of the EPA side and website, and as you can see here, none of the funds that are currently being given for waste prevention strategies are virtually targeting biomaterials. And of course, this is just one example that we brought, but we found the same pattern across many different government agencies. So ultimately, why this is important is because at the moment, there are $715 million being invested in waste prevention strategies. However, none of them are currently targeting biomaterials. And so ultimately, what this shows us is that biomaterials have yet to be positioned as waste prevention strategies. And so at this point, we stopped for a second and we asked ourselves, okay, do we really need more biomaterials? Do we want to keep pursuing these? Or do we want to help existing biomaterials actually leverage and scale to the point in which they can achieve greater impact? And so the reason that we're here today in the Biodesign Challenge is not because we have a biomaterial innovation or a product of our own. The reason we're here is because we're building a platform for different biomaterial innovations to showcase their impact.
So as Lucy walked us through, by now we have seen waste prevention matters. We have seen there's a specific subset of biomaterials that can have an impact on it. And lastly, we have seen that there's a big opportunity out there to drive investment. Then the question becomes, what's missing? We need to be able to show impact. And for that to happen, we need to be able to obtain data, because you cannot measure what you cannot track. Now, one of the biggest challenges associated with tracking waste prevention data is the fact that it's hard to see and measure something that doesn't exist, aka measuring absence. With recycling, we see bins filling up, trucks collecting, and facilities processing these recyclables. It's tangible, it's traceable, and therefore measurable. Waste prevention, on the other hand, is about the absence of waste, and it's invisible. And that's exactly what we are doing with our project Awayste, making the invisible visible. By combining biodesign and data, we want to provide insights to policymakers, businesses, and investors on how biomaterials can impact waste prevention, with the hope of raising awareness and driving investment. But to achieve this, we must first answer one crucial question, and it is, how much packaging waste is actually being prevented by these no-waste biomaterials at the first place. And our impact tracker provides a clear answer by displaying the amount of waste prevented by these innovations to bring visibility to the current state of things. But we are also aware of the fact that this is not currently a huge market yet, and that is exactly why we think it's more important even to showcase the future potential of these biomaterials. And to show the potential impact, the question we need to be able to answer is that how much impact would it have to implement these solutions at a scale? This is where the impact calculator comes into play. For example, it would show us how much waste we would prevent if we replaced all the plastic shampoo sachets currently that are on the market with the bio-based water-soluble alternatives. Of course, there are many other ways to measure impact, but we are currently focusing on financial aspects such as waste management cost savings to demonstrate the potential of investing to these solutions from a governmental perspective. Then once we have like a clear understanding of the impact and how it is achieved, you might want to know, okay, who is driving this change? And the solution provider directory offers information on who are those companies and what are their products that are driving that impact. You can filter the directory in various ways, such as industry, packaging types, material types, and even prevention strategies as well. In the end, with this platform, our goal is to paint a picture of what the future could hold. And this is precisely the information we wanted to have when we started our own biomaterial project, but we couldn't find anywhere. And through this project, we hope to answer 3 key questions. First, how much money do we need to invest to these biomaterials? Second, how much waste could we prevent with that investment? And lastly, what impact would that bring from an economic, social, and environmental perspective? So we know that Awayste is a first effort of its kind to make waste prevention visible, but we are optimistic about its potential because of the other successful and similar initiatives in other industries that we see, as you can see in here. In fact, the design of our platform was inspired by RIFAT, a non-profit in the food waste space. So these precedents give us confidence in our approach, and our goal is to become the reference point for the waste prevention and biomaterial industry. And currently, we are focusing on no-waste biomaterials, but we envision expanding to other biomaterial categories in the future as well.
So we began this presentation with a very big claim, that biomaterials can actually impact the waste crisis. Now, in order to do so, biodesign can not only be instrumental. We must bridge biodesign, data, and policy. And the data that we have today is actually scattered. It's hidden within individual efforts, and we are acting in silos. So we are here because we want to bring this data together. But in order to do so, we need your help. So if you know any biomaterial companies, people in academia, or policymakers that can help us make the case, please send them our way. Now, today, we want to leave you with one last message. Whatever we do and however we choose to make an impact, we must remember that we are part of a collective that can pave the way away from waste. Thank you so much.
Hello. Hello.
All right, Hello? Hello?
Yeah.
Is this working?
Yeah.
Oh, okay. All right.
Thank you for that excellent presentation, great storytelling, and I love the approach to kind of finding ways to support the businesses that are already operationalizing their biomaterials. In the product that you showed us basically a dashboard that provides a snapshot of impact right now. Yeah. I'm curious if you've explored additional services that your platform will offer, probably to industry who wants to adopt biomaterial waste reduction. Yeah. Right? Are there ongoing things that you can do on this platform beyond that one-time snapshot?
For sure, see, definitely. I think, of course, we have great vision for the places this could go. I think initially we have thought this more in terms of impact, but definitely we want to tie this with policy making, especially in the context of the EPR, which is the extended producer responsibility that's now going to begin in the United States. We think this tool can have great impact for that. Of course, we see that there's a route in which this can be used by the private sector, because more than that, it's also a search tool in which they can find replacement or alternatives for what they're looking for. And actually, yesterday, we received some pretty interesting feedback about maybe thinking about how the people or the end customers can also interact with that. And one of the things we have been discussing is, as a beginning stage at least, would be labels, to ensure that we are clearly communicating that no waste aspect that we're trying to push for.
All right, next questions.
Again, a great presentation. Did you do any research into how consumers interact with these products? Is that a pleasant way to drink? I've not tried that kind of packaging, but did you get any feedback on that?
Yeah, actually one of the research that we did when we did our biomaterial, our poly bag, is the fact that we figured out actually when it becomes, the packaging becomes the product experience, people tend to have more leaning towards to use that packaging even more. They are getting excited about it. So this is also one of the, I think, motivational points that our project is also trying to connect through a user's perspective as well.
All right, next question.
This really interesting project. I guess a question about sort of, this seems to be looking at sort of the macro scale of waste generation, and waste management is more of kind of a micro scale at the local government level. Can you talk about how you are going to sort of, are thinking about integrating sort of local waste management sort of systems and government decisions into your platform?
For sure. So I think one of the most important things for us is that we hope that this is going to be a very critical space or platform in which we are actually able to assess the reality of what we're doing. So I think why we're focused on no waste biomaterials specifically is because we want to keep reminding people that making no waste is actually what we should all be aiming for, you know? So yes, it's okay that we are finding better materials to do cups or to do compostable things, but we need to remember that like producing no waste is kind of the top tier priority for us. And so I think by encouraging that mindset, it's also a way in which we can start interacting with the government, because one of the things that bothers us the most is that there's a huge industry in recycling. People are actually making a living out of this. What we want is a world in which no waste or preventing waste is an industry itself. I mean, we live in a capitalist society. This is kind of the rules of the game that we have been dealt with, but we envision, or what we dream of, is kind of building an industry on making no waste. I don't know if that answers the question.
All right, I think that's all the time we have for questions, but thank you so much, Parsons Awayste team. Great presentation. Okay, next could we have the CCS Salt Mines team please come up.
Hi. Okay, so I'll get started. So, due to the increase in salt in our environment, we could be seeing an extreme shift in color in our ecosystems. This is a potential future not only for Detroit, as represented here, but globally. This is due to freshwater salinization syndrome, which is the global rise in salinated environments, which has systems-wide impacts, and which is the center of our project, Salt Mines. So we are a group of color and materials students, master's students from College for Creative Studies, who are exploring this complex and relatively new field of research through color awareness. I'm Vivian Zhang. Thank you so much for the opportunity to present this once more.
So we wanted to start off this project with grounding ourselves in shared values. One, the preservation of ecosystems. Two, utilizing this platform for collaboration and community development. Three, expressing emotional systems through color. And lastly, human safety and harmony with all life forms. We started our research in our own backyard. Detroit is a city built on industrial success, driven by the automotive industry and our natural resources, such as fresh water. But there has been a price for that success, leaving us with unusable land, environmental racism, and polluted waterways. But of course, this isn't just a problem in Detroit, it is a global issue. Every day, 2 million tons of industrial waste is released into our waterways across the globe. What we found is that salt is a common pollutant, driving the increased salinization within our waterways, also known as freshwater salinization syndrome.
So because we wanted to focus on salt as a pollutant, we wanted to learn what salt was outside of our basic understanding. But did you know that salt is not actually a biomaterial? Because we did not. But we found out that it is vital for the survival of humans, animals, and plants. In humans, for example, salt aids in the conduction of nerve impulses. We also found that there is a very careful balance, as salt is part of geological and hydrological processes. However, due to human activity, salt has become devastating in excess, contaminating our drinking water, damaging infrastructure, and endangering and killing wildlife.
An example of excessive use of salt is road salt, something that we love in the US and especially in Michigan. It keeps us 78% safer on the roads, but it comes at a cost. We as Americans spend $5 billion annually just on damages for vehicles and infrastructure. And the sheer amount of salt we're using not only affects infrastructure, but it also threatens lives. There's the tragic example of the Flint water crisis, where the water there was affected with lead 1,000 times over the safety limit. We were surprised to learn that this was in due part to road salt runoff, which salinated the waterways, which then corroded their pipes, which then leached lead into the water system, poisoning the community of Flint. And this was very complex for us to, sorry, it's okay, for us to understand. So we decided to consult an expert panel. So we have Abby Heilman from Saltwatch, Carrie Kofer, who is a color master, and also Erica Gardner, who is our sustainable systems expert.
We then began the process of experimenting with salt. We started looking at how salt interacts with water and different materials. We observed how the cubic crystallization structure formed. And for example, it grew more on cotton than it did on hemp. We also looked at how salt interacted with metals such as aluminum and copper, and how they dulled in appearance and corroded.
Okay, so after observing salt as a material, we want to understand its presence in our local environment. So we visited the Detroit salt mines, Belle Isle, and the Rouge River to test for salinity through conductivity and chloride levels.
and what we found left us very disheartened. Our conductivity findings for the Rouge River far exceeded what was reported by the Environmental Protection Agency for that region. And this sounded an alarm bell in our heads, making freshwater salinization syndrome even more of a reality. This is real and this is bad, and we need to be focusing all of our attention on it.
So through this overall process, we just wanted to take pause and go back to our values that we had originally set out for ourselves. And like Smita said, we were like, okay, we really need all hands on deck for this global issue. So from there, we kind of convened and said, okay, it's not necessarily going to be a single biotech solution. It's too complex and across so many systems. Instead, we're going to need a collective mindset shift. And so in order to do that, we broke this up into 5 different roles. Those who mobilize thought, those who connect the dots, those who strengthen bonds, those who uplift voices, and those who build the structures. And all of these being equally as important in the prevention of freshwater salinization syndrome.
So to avoid this extreme future, we need a system that unifies. Throughout history, color has been used as an indicator for environmental evaluation. With this in mind, we selected the Munsell color system because it is considered one of the most scientific color measuring systems. An example of this is the Munsell soil classification chart, which is globally recognized in soil research. So we asked, what if this system could include freshwater salinization syndrome? Using this as a tool of color evidence with the potential to map out the impacts of FSS.
To visualize this potential addition to the Munsell color system, we broke up freshwater salinization syndrome's impacts into 5 colors. The first being chemical cocktail blue, which represents how salt mobilizes metals and contaminates water and soil. Next, we have Ghost forest green, representing the sickly green color that plants take on when their nutrients and oxygen are choked out by salt. Then we have halophile red, which represents the bioindicator of halophiles which thrive in saline waters, turning it pink and red. Of course, the corrosion power of salt creates corrosion orange. And finally, we have aerosolized gray, representing crushed salt particles polluting our air. The color representations of freshwater salinization syndrome highlight the severe impacts that can occur if we don't collectively come together to address this issue. Thus, we foresee the need for an exact measurement system, such as the Munsell color system.
But of course, we don't necessarily want to design for the most extreme future. We would like to try to propose this as something preventative first. So as color designers, we are kind of taking on the role of the mind to mobilize thought and using color as a tool. So consulting Carrie Coffer, our color master, she says, the Munsell system aids in the identification of the differences of color and allows for engagement. So we present this to you today to ask what role you can take on to prevent freshwater salinization syndrome. Will you, like us, mobilize thought, creating the frameworks for certain databases, research, and measurement systems?
Will you identify system-wide connections to reduce salt pollution? Or will you build relationships between various sectors to ensure this is a top priority?
Or will you innovate new design and construction to prevent further damage to our built environments?
Can we stop freshwater salinization together with our collective salt mines? Thank you.
Thank you for the presentation. We'll take questions from the judges now. All right, Laura.
That was a great presentation, and I really enjoyed seeing the cards in the exhibition last night too. And you asked some really great questions at the end and connect them to the cards, and I was just wondering if you did any testing with users and kind of saw how people might interpret them and understand their environment through the cards?
We didn't have a chance to actually test them before this, but last night was a great experience to meet all of you guys and have a chance for you guys to we did it in a more playful manner where we asked people to kind of gravitate to which card they were interested, just by color, as a way to kind of open that gate of like, oh, well, you've selected red, so that means you must be very good with relationships and you can form bonds. So just as a way to open up that conversation of how you can get involved.
Hello. Thank you for your presentation. I have some questions about community engagement and how you see yourselves unrolling this initiative. Would it start locally in Detroit? Would you start with environmental groups? Have you thought about interacting with the EPA at all or other government agencies to adopt this color method?
Okay, so we are in close contact with Abby Howellman from Saltwatch, and so they have there are 2 large annual salt conferences every year, and this is like their biggest one yet this year, she told us. But our ideal would be to be able to bring together to join these groups of researchers and salt-minded people, but also bring our perspective of color to kind of create more of to open up this research more to like the everyday person like ourselves. Did I answer the whole question? Okay.
Hi. I'm wondering if regionally you've identified perhaps like another community that's close to you that might be doing this kind of work, or if you've like chosen people around your communities around you regionally that you could partner with who are also experiencing this issue.
So freshwater salinization syndrome is a global issue, but we haven't had a chance to go out and partner with anyone yet, but we'd really like to. So far, our project is just kind of centered around Detroit. Some of all of the samples that we collected were from Detroit and the surrounding area. The salt that we had for Chemical Cocktail Blue in our display was actually collected from the Detroit salt mines. But for now, we haven't had a chance to interact with our community members outside of the lovely people of Biodesign.
Who would be your dream first partner to work with, and what would that implementation of this color system be?
It would be Sujay Kaushal. He is the lead researcher in the field, and he's we call him the father of freshwater salinization syndrome, because he coined, along with other researchers, coined the term freshwater salinization syndrome. So if he is watching, will you please respond to our emails? Oh, yeah. Okay, so just to kind of add on also to the previous question, back to the question about if we're going to partner up with EPA or things like that, it would be some sort of conference where all of these groups, through all these roles, uplifting voices is about putting forth public policy, but also of course designers are needed, just kind of bringing in all these minds. I'll just give you one really quick example. Abby Howellman told us that at her house there's this awning, and if it just extended 1 foot farther, then it could save her I don't remember the exact amount of salt, but a ton of salt each year, just if it had that one design element change. So kind of being able to bring together all of these different thinkers to take in these considerations. But as a measurement tool, of course ours is a representation of how we could see this tool going forward, but it would be, if we were working with actual researchers to be able to make it more accurate, then that would be wonderful. And accessible to people as well, because the Munsell system is something that you can only have access to through purchasing.
All right, I think that's it for questions. Thank you, CCS Salt Mines team.
Okay, so could we now have the Universidad del Istmo team, Kanbe, come up to present? Here they come, all the way in the back.
Hello, good morning, everyone. We are Hearth. I am Evelyn. These are my teammates, Ali, Jime, Gabriel, and Anna, who sadly couldn't join us today. And now we are going to present the story of Olmeo.
Olmeo Sagasume is a Guatemalan farmer who raises chickens and faces the daily challenge of protecting his crops. These crops not only support of his family, but also serve as food for his chickens. Olmeo lives in the rural area, just like 46% of country's population. Is there another one? This one? Yeah. Okay, yeah, that works. Yeah, so like I told you, Olmeo lives in the rural area of Guatemala, just like 46% of country's population. Olmeo, like many others in the rural area, supports his family with corn. Corn crops are crucial in Guatemala, occupying more than 10% of countries of the territory. One day, Olmeo bought corn treated with pHen, an effective but dangerous pesticide. Unaware that it wasn't safe yet, he fed it to his chickens. The next day, he faced a devastating scene. His chickens, a crucial source of income for his family, were dead. The pHen had poisoned them. So that led us to the question, what is pHen? pHen is a strong chemical often used to protect stored grains. It works by releasing the gas, eliminates insects, but the problem is that it's highly toxic. And this is not a problem in Guatemala, because pHen is the most widely fumigant in the world. Let's talk about the process of pHen. So the first part is harvesting the corn and letting it dry. With the corn dry, it's placed in bags or silos. The pHen peel then is placed in a way that it doesn't touch the corn. When the peel contacts air, it starts releasing the gas after about 4 hours, spreading and killing pests like weevils. The corn is considered safe to consume after 3 months. After the peel process, farmers place the pHen in the soil, liberating the gas in the environment. The phosphate gas during the process can severely damage respiratory and nervous systems. In the rural area of Guatemala, due to the easy access and lack of regulation of pHen, is used for suicide. As well, damage the environment, contaminating soil, water, air. Olmeo case is not the only one. Made us reflect on the widespread impact of pHen and what are we actually eating.
And this got us thinking, why risk our life using pHen when we can find a solution in nature? And while using innovation, biodesign, and the respect for nature, we proposed a new organic pesticide to use. And we created Cambe. This is an organic pesticide. We use Argemone mexicana seeds for the repellent properties. We just called it AM for short. And propolis. These are our 2 materials. And now you may be wondering, why focus on protecting corn? And I'll give you the context real quick. Corn is also known as the golden grain in Guatemala. It is a really important part of our culture. And according to the Popol Vuh, this is a sacred book of the Mayan culture, the first people were created with corn. And this just created with us such an intimate connection between humans and earth. corn is more than a crop. It is a manifestation of identity and belonging for us Guatemalans. And we eat it every day in the form of tortillas, tamales, and atoles. As you saw, corn is the main source of income for many Guatemalan families. So it is really important for us to create something that protects corn, but also people. And by creating Cambe, we're just not protecting corn from the pest, like the weevil, but also honoring our cultural roots. And getting into the real deal, how is Cambe composed then? As I mentioned, we use AM seeds and propolis to protect the corn. And just get a little bit into that. We use, propolis is formed from many substances. It is essential oils, pollens, balsams, resins, waxes. This is a bee product, but we're just focusing on the antimicrobial activity that it has. And now let's talk about AM. Argemonia mexicana, in a study did by la Universidad Autónoma del Estado de Morelos, they concluded that it has a 98.9% of mortality against the weevil pest. And it reduces to 0% the development of new generations of weevil and grain damage.
So now let's talk about the experimental process. Our main goal was to obtain favorable results that demonstrate effectiveness of the properties of propolis and AM seed investigated against the weevil corn pest. So the samples are as follows. We have 1 pound of corn. Then we have 4.5 grams of AM seed. And here it is important to note that we decided to use AM seed, ground AM seed, for better result, so the properties can go through the sacks. And following up with 5 grams of propolis. So let's talk about that we aim to recreate the experimental conditions as closely as possible of how they will be carried out in real scenarios. Our main goal was to make the bags as modular as possible, so we use a pound of corn that shows and demonstrates to work as a modular measures of how will be carried out in real scenarios. So, as a result, within a week, beneficial outcomes started to appear. In the samples with AM seed and propolis, dead weevils appeared. That's great. And in the other hand, those without alterations had more corn weevil eaten and more weevils, so that's a great point. So, following up on how those Cambe works then, based on our research, the average family in Guatemala harvests around 600 pounds of corn. Yeah. And between October and December. As mentioned earlier, Cambe's idea is to make it as modular as possible. Therefore, we thought of producing it in boxes that can cover up to 100 pounds. So 1 box covers 100 pounds. It will concentrate a pound of a product divided into 2 sacks that has half of the pound between. And a package of Cambe covers 100 pounds. And we also think about ahead, and we decided to think of, in Guatemala harvest around 600 pounds, so we made a presentation that meets these needs.
So what is expected from Cambe? By developing Cambe, we are paving the way to our multiple benefits that include crucial aspects for our society and environment. First, from a cultural perspective, this product represents a return to the roots and ancestral traditions of cultivating in harmony with nature. In Guatemala, corn is more than just a crop, and this method honors our heritage and respects the knowledge passed down through generations. From a natural perspective, the benefits are significant. Unlike phostoxin, which introduces toxic chemicals into our environment, Cambe is safer by the use of natural ingredients. Although effective, phostoxin poses serious risks for farmers and their families, as its misuse can lead to severe poisoning and death. Its easy access, low price, and lack of self-regulation makes it lethal for humans, leading to its use as a method of suicide in rural areas of Guatemala and many places worldwide. By eliminating phostoxin, we are protecting the health of our communities and ensuring that the food that we consume is safe for consumption.
And now, did we mention that we're all architecture and design students? So it has been a challenge from the start to think about something outside our field, but we never limited ourselves to just think about something architecture related. And coming across the case of Olmeo really made us reflect about the priorities that the Guatemalan citizens have. And last but not least, we would like to thank the Biodesign Challenge for inviting us, la Universidad del Istmo, our Dean, Ana María de Garcia, for her unconditional support, Alejandra Sierra for her assistance in this project, Juan Pablo Arrocha for introducing Biodesign Challenge to us, and architect Roberto Sosa for being our tutor these past few months. Thank you.
Thank you, ISMO team. We'll take questions from the judges.
Hi, everyone. That was a wonderful presentation, especially in terms of the cultural connections that you made. But I'm interested to know if during your process you had a chance to think about the kind of pushback that a product like this might get when it goes on the market, when the giant corporations that are selling the toxin and making lots of profits off of it realize that you're presenting something that people would want that would decimate their market. They're powerful. Did you have a chance to think that through yet? Because you were really thinking about the culture, so.
Yes, thank you. Excellent question. Yeah, we had the opportunity to interview a lot of farmers, and we also have a little bit of connection, and we talk with many organizations in Guatemala, such as the MAGA, it is the Ministry of Agriculture, and they were interested in the product because right now in Guatemala, phostoxin is all the people know. They use it because it is something that it is used from generation to generation. And now introducing a new product, and especially an organic product, we are changing that mindset, and they were interested in that, the farmers and the organizations.
Yeah, that was really great. I guess I wanted to ask, is the idea that the seed that you're introducing going to be part of the grain, or is that going to get separated out during the actual, when someone's actually eating it? Because I think that the material itself is also has some level of toxicity, right? It's very similar to mustard, and it's not really used as a, animals don't eat it, for example, because it's toxic to them.
Yeah, we thought about it, and as you saw on the presentation, it works separately. We put the product in the bags inside of it, and all the properties run through air. That's like the idea that we had. Especially, yeah, because we don't want it to be touching the direct corn.
I have a question over here. I was curious if you can dive deeper into your iterative design process. You presented like a nice finished product, but tell me more about the process. Putting this beeswax with the seeds. Did you test just the flowers? Did you understand the impact of these separate components? How did you come up with the recipe? If you can share more about that, that'd be great.
Sure. Yeah, we came a long way. We tried many different products. This was not the first idea. Propolis was the main idea. We really wanted to use it, and now we are using it as an antimicrobial barrier. But, at Hemonia Mexicana, the doses that we're using in our experimentation It was determined also by the university in Mexico. They use 1 gram of Argemone mexicana to treat 100 grams of corn. And that's the proportion that we use. That's what we use, 4.5. And yeah, we tried many different another organic materials. We tried, in Guatemala we call it acacia, but it didn't work out as well as Argemone mexicana did. Yeah, we tried.
All right, that's going to be all the time we have for questions. So thank you, Convey team. Okay, because we're running. Yeah, could we get audio? Hello? No, this one's off. Thanks though. We're gonna just take a quick break because we're running a bit short on time. So, feel free to stretch, stand up, stretch your legs. We'll start the next presentation at 11:30, and that will be UT Austin, Consuming Our Consumption.
Hello? Okay. Can we have everyone settle, please? Clap once if you can hear me. Clap twice if you can hear me. Please sit down. Thank you very much. Awesome. I know people are gonna come trickling in, but I want to keep us as close to not super late as possible. Okay, cool. That was our first half. We're gonna go into our second half, our last 4 top 8 teams. Up next, we have UT Austin, Consume Our Consumption. AV, she's going to use one of the handhelds.
Hello? Hey, okay. Hi, my name's Anya Caney, and me and my partner Anon have Consume Our Consumption from the University of Texas at Austin. Has plastic pollution got you feeling stressed? Overwhelmed? Guilty even? The environmental crisis is such a bummer. Plastic is a ubiquitous contaminant that pollutes our environments and affects the health and ecology of all of our environmental systems. The 1950s marks the start of the Anthropocene, or the geological timescale that is defined by the layer of plastic that will forever be a part of our planetary history. Every single year, humans produce over 430 million tons of plastic, and 380 million tons of these single-use products will become waste. This massive pollution stream is choking our wildlife, polluting our groundwater, and poisoning our soils, overall impacting the health and safety of all living organisms. But worry not, the solution is here. From our company EcoBite, we present placetamol, the little green pill. This novel drug gives any organism the ability to eat, consume, and digest plastics. And that's right, ladies and gentlemen, this includes you. Placetamol also provides relief from other common ailments, such as eco-anxiety or dietary sin. It really is the miracle drug that we've been looking for. But how does this work? Well, first, let's get into what plastic actually is. This is polystyrene, also known as styrofoam or a common plastic. And if you look at the top there, you'll see a bunch of carbon atoms all connected together. This carbon backbone, as we call it, is present in all types of plastic. These bonds are really strong, and that's what makes plastic so strong and durable, and also so resistant to biodegradation. They're very hard to break. This is where these guys come in. There's worms in here. This is the superworm, also known as the Phobos moreo, and they are incredible. Their digestive microbiome contains bacteria that has the ability to break apart that carbon backbone. So as the worms eat, their digestive tract is turning plastic into carbon dioxide and water. That's pretty incredible. Furthermore, they're actually metabolizing and using plastic for energy, not just breaking it down into smaller parts. However, the current study methodology is outdated and honestly needs improvement. If you want to study these plastic degrading microbes, you would take a worm, slice it open, and remove its gut for further experimentation. This not only reduces these worms to single-use organisms, but it also totally limits their academic potential. By killing worms with observations, you prevent the ability to track changes in bacterial effectivity over time or across generations. So you can't see how these bacteria change inside an organism. Furthermore, it's limiting to the ability to select for more efficient plastic degradation, which again, if we're fighting a global pollution crisis, we want the best we can get. This project introduces a new way of studying these worms. Instead of taking bacterial samples from their extracted gut, I use samples from their frass, or worm poop. So this project experimentation is all about finding a way to take bacterial cultures from their frass to expand the potential for environmental remediation of these organisms. The key idea is that as the poop or the frass travels throughout their digestive tract, it gets covered in all those plastic degrading microbes. When it comes out the other end, I can take bacterial samples from that. So here are the worms. Some of them are fed plastic, some of them are fed carrots, but it's very important to note that all worms, all superworms, contain that plastic degrading bacteria in their guts, not just the ones fed plastic. These carrot-eating control worms are more to provide a behavioral standard to ensure that plastic ones are truly thriving on their diet. So this is an example of some plastic degradation they've done, and as you can see, they're really good at what they do. In just 6 weeks, 200 worms were able to consume about 4 grams of expanded polyethylene, or plastic. So in order to collect their frass, this is what the setup looks like. They essentially get a little vacation in a beaker overnight, and I get to collect bacterial samples without harming any of these worms. So here's some data from a collection, and here's what their poop looks like. I collect all sorts of data on the collection process to further optimize and get the most out of these worms. So here's some raw data, and here's how I would use it to increase the productivity of this experimentation. Okay, so once I have those frass samples, it's time to take bacterial cultures. Firstly, the frass or the poop is gonna go through a couple rounds of cellular washing and centrifugation to remove all of the not fully digested plastic, any internal shedding, anything that isn't bacteria. And this yields what is essentially a solid lump of bacteria. This will get thrown in a nutrient media to turn a little into a lot. So if you look at this media and you look at all these chemicals over here, you will notice there is no carbon in here. So the media contains everything essential for life except for the most important ingredient. Recall the carbon backbone present in all plastic. This is a polystyrene nurdle. It's basically just a plastic bead, and it also gets thrown in the media. This is the sole carbon source for the bacteria to eat. So this prevents any growth from anything that can't degrade microplastics. Pretty cool. Here's some cultures. Here's some cultures under a microscope. All that turbidity in this provides examples of there is bacterial growth in these cultures, which is super, super positive. And then this is just an example of some contamination I ran into, just to show that this is a constant trial and error process because it's a developing methodology. So, yeah. All right, so we have these plastic-eating worms. That's pretty cool. However, like I said, we're fighting a global pollution crisis. The worms are just not gonna be enough. However, their bacteria in the frass, that's what holds the key. What if we could take this bacteria, this plastic-degrading microbial activity, and transfer it to the guts of other organisms? Plastic-eating dogs, sea animals, even algae and fungi. Through methods of bacterial transfer, such as fecal matter transplant or probiotics, we can introduce this plastic-degrading bacteria into other gut systems. If eating is consuming and plastic is our consumption, then this is consuming our consumption. First, let's get into the food systems we currently have. Our current food systems are inherently flawed, and these problems need to be addressed. For one, there's inherent power structures in our giving and receiving food. No one makes their own food, and this creates inequality. Furthermore, the globalization of the food production process has led to this incessant desire for exotic and tropical foods. This places a huge strain on our environment from transportation, over-farming, and other environmental factors. And furthermore, is dietary sin. Everything you eat to sustain your own life had to be taken from another life. You cannot eat without killing. Eating plastic solves all of these issues. For one, everyone has access to slow and local food. And furthermore, we're not just eliminating, but actually reversing environmental violation that we've done through over-farming. By taking plastic out of these environments that we've damaged so heavily, we're allowing the earth to reclaim its natural beauty. And then furthermore, by eating plastic, you're not killing anything. So you get this self-cleansing of that dietary sin. So placetamol, it really does have the solution to all of our food issues. So let's take a moment and explore this future that EcoBite provides. Imagine cows that graze on landfills instead of pastures, birds that are energized by the plastic they consume daily instead of choked and killed by it, sea animals that are able to reclaim their environment that we have made our dumping ground for decades. These microbes hold the key to reclaiming this environment. Forests will reemerge, the oceans will be cleansed, and even your neighborhoods will be cleaned up from all the plastic debris that's littered quite literally everywhere. And it's not just the animals that get to partake in this plastic eating revolution. People too have the ability to participate. With just one pill a day, you'll be able to consume as much plastic as you want. You can eat up all the waste around you, clean up your neighborhood, and clean up the ocean. And if you're a terribly selfish human being and this environmental call to action just doesn't appeal to you, there's an individual incentive for each one of you. You know all those microplastics that are, floating around in your blood? No more. Plastic can never get past your gut because that's where the degradation is taking place. And this is just the beginning of the utopian future that EcoBite provides. No more grocery shopping. Plastic is everywhere. Making a meal for your family, it's so easy. Solving world hunger, it is so easy. And no more greenwashing from these shady politicians. If he really cares, he'll be eating his plastic. Imagine fancy dining experiences, plastic eating theme parks, culinary tourism to the Pacific garbage patch, and even plastic eating cults. You can release all of your desire. Eat as much plastic as you want with no limitation. We can reclaim our environment through plastic eating and EcoBite, eat our way out of plastic pollution. So we can eat plastic. What's next? Placetamol is a solution to plastic consumption, and it is therefore designed to eradicate its own need. However, this isn't the first miracle solution that you've been sold. Every so often, you see these solutions pop up. We're going to cure cancer. We're going to solve the environmental crisis. But a lot of times, it's plastic and oil lobbyists that are selling you these solutions, and it's just to really sell you more plastic. I mean, think about it. By all this recycling, it enables even more production of plastic, and more production of plastic means you have more to consume. There's more to sell you. It enables endless consumption. By increasing our capacity to recycle, we increase our capacity to produce. And in turn, that increases our capacity to consume, and then recycle more, produce. There's a loop. So would EcoBite really just create this dark, infinite loop that only enables the very systems it set out to destroy? Is this a future that we would even really want? So yeah, this is EcoBite from me and my partner Anan. We are from the University of Texas at Austin, and I would love to thank Jabal Lee for my biodesign class and lab this semester, and also the bioprospecting stream at University of Texas at Austin for the research.
Judges?
Is plastic fattening? What is its nutritional value?
Just carbon, so it's pretty good for you. The worms are very happy on it.
Yeah, I have a question related to the texture of the plastic that you propose to eat. A lot of the plastic is hard and difficult to chew on it, so like, would you have a solution for like, I don't know, so it brings into like cooking or preparation of the plastic for consumption?
Yeah, cookbook coming soon. No, so the worms that are our model organism for this bacterial degradation, they can only chew on soft plastic. So think like styrofoam, or this is expanded polyethylene, to do that mechanical degradation. That's why taking out the bacteria from their gut is really key to harnessing this plastic degrading ability, because the microbes themselves can colonize the surface of hard plastics. So like the plastic nurdle, the polystyrene nurdle that's included in the cultures, they'll colonize the hard exterior of that and extract the carbon that way.
Do the worms need other nutrients besides like carbon? Are they eating other things other than plastic?
They don't need other nutrients. Worms have actually been proven to thrive, or superworms specifically, have been proven to thrive on a solely plastic diet. However, there are studies that show that including bran or oats in their diet, just even a little bit, will increase their ability to eat even more plastic. So I also have bran in their diets just to, again, make it even more productive.
Dan, and then Elaine.
Thank you. So you end your presentation with sort of a disclaimer of sorts. I'm wondering if you can kind of describe to the audience what the research that you're doing, what you're actually proposing, how you would actually use this in the world.
Yeah, so the research I do, again, through the lab at UT Austin is, guys, we're not actually making a pill. And it's actually still very early on in the process. But right now, it's all about identifying the bacteria present in the gut samples. And what's really interesting about this project specifically is instead of taking the cultures from the guts, where we know there's Klebsiella, Citrobacter, certain plastic degrading microbes, there might be entirely new microbes in the frass. And so like when I get back, I'll be focusing on identifying what the actual plastic degrading bacteria is. Once we identify those, we can amplify it and put them to all sorts of use.
Hello. Hi. So I have a question about scalability and the relationship with like the worm's actual life cycle. I began a question like that yesterday, but could you talk about like impact in terms of like how many worms are we talking and how much plastic?
So the worm's ability to degrade plastic is limited just by their very small creatures. And plastic actually doesn't spend enough time in their gut with those digestive microbes to be fully degraded. So a lot of their frass will be contaminated with microplastics. That's another reason why extracting the bacteria and using just the bacteria for plastic degradation is really key to be able to harness this power fully. These worms specifically, it takes, based on limited data, I think I've had about 4 bricks consumed. They'll eat about 0.1 milligrams of plastic per worm per day. This scales up to about 200 worms take anywhere between 4 and 6 weeks to consume like a 4 to 5 gram brick of plastic.
Thank you so much. Well done. Can we, yeah. Can we have the Moholy-Nagy University come up next? Fun.
Hello. Hello everyone, we are the team of pHen, and we would like to introduce you to our project, One pHen, Infinite Use. We are coming from Budapest, Hungary, Moholy-Nagy University of Art and Design. We are quite a diverse team as we are coming from different fields of design. Baldi here from media design, Sonia from graphic design, and myself, Nori, from textile design. But these differences have only benefited us throughout the work, as we all have different skills and knowledges. But there is one thing that connects us all, which is the importance of color in our professional lives. Colors are not only important to us, just us. Colors are the mother tongue of subconscious, says Carl Jung, psychologist. Colors are everywhere in our history, culture, creativity, and everyday lives. Colors catch the eye and represent our personal style. But what is the cost of our devotion to colors? There is one tool that is represented in all of us' lives from our early childhood, and these are pens. Did you know that 10 billion plastic pens are thrown away each year? And this amount could fill 1,467 school buses. Now let's look at the most important components of pens, which are inks. Inks contain volatile organic compounds, solvents, heavy metals, and other chemicals. And they are extremely harmful for the environment when they are disposed of improperly. And additionally, they can cause behavioral abnormalities such as hyperactivity in people. So we started asking ourselves, how can we create a sustainable alternative that is fun and engaging so people would actually use it? Our goal was to reach people who are not primarily interested in sustainability, but still make them use a sustainable tool. Do you remember those pens which we used as kids with multiple colors? Well, we were inspired by them and started examining ways how can we create an ink that can change its color. We found out that there are certain pigments that can change their color due to the change of pH. And these pigments are coming from different sources, which we have paired to different levels to make the ink based on age and biochemical knowledge. Let's look at level 1, which pigments are coming from fruits and vegetables. These pigments can be, for example, anthocyanin, betaline, curcumin, or polyphenol. Level 2 pigments are coming from bacteria. For example, J. lividum can change its hue due to the change of pH. It can change between a light purple to even a blackish purple. Or Streptomyces color can change its color. As bacteria do not have that wide range of colors as fruits and vegetables with anthocyanin have, research papers suggest that they can be genetically modified with anthocyanin. And these changes have been achieved, for example, with E. coli or S. cerevisiae.
As most new eco-friendly solutions target businesses in this era, we target individuals, especially designers and educational purposes. Based on our research and conclusion, our solution is Splan. One source and multiple colors. And the best thing, you can do it at home without causing any harm to the environment. But for proof of concept, we looked up different kind of pH reactions we could get with household chemicals. But colors are not enough. We need the right viscosity, so we looked up how different kind of pens and markers work. For our idea, the viscosity of markers work the best. So now let's see in practice. We found out that anthocyanins are solvable color pigments which can be found in wedges, and due to the pH level changes, they may appear red, purple, blue, or even green. As Nori mentioned earlier, here you can see our range of colors of wedges. But red cabbage is one of the richest in anthocyanins. We tested it with citric acid and baking soda and made a solution which was highly pigmented and had the right viscosity as well. Here you can see our range of colors. But for our more advanced level colleagues, let me show you our experiment with the bacteria, the J. lividum, which has a unique purple color, as you can see. During our experiments, we had to figure out how to create a successful, stressful environment for the bacteria to show its color and to how to solve the problem of contamination. The question was how to create the never-ending ink. For example, we tested it with extracting with alcohol or even evaporating it, as you can see. But the best method was growing the never-ending ink, was just creating a thicker liquid culture. But now let me show you the method of how you can create the cabbage ink, not just because we love cabbage, as you can see, but it's really easy and you can make it at home even tonight. It's very straightforward. You just need to dry your vegetable. After that, use a grinder to make a powder, add water, and then use a sieve or a cheesecloth to extract the pigmented ink. So both bacteria and cabbage have a great potential. pH-reactive bacteria is self-regenerative, so no agricultural land needed, and you can use on synthetic materials. But on the other hand, it's a much slower process to make, and it can die if you are not careful enough. On the other hand, cabbage is very easy to make. It's really pigmented and have a good chemistry with citric acid and baking soda. One con on its side is the additional smell, but during that time we became to love cabbage.
During the rapid prototyping phase, we took two main approaches. The first one was bacteria as a source of pigment. Now, working with a living organism posed a lot of challenges. For the prototype, we wanted to create an item that could be sterilized by heat, but that also meant that we couldn't use conventional 3D printed materials such as PLA or ABS, but rather use those materials to create a negative mold to be filled with silicone. While we were working with bacteria, we realized for our final prototype, this could not be the source of pigment because of its hard to access by the community, and also outside of a laboratory environment, it's hard to recreate the steps needed to create pigment. So we took all our learnings and for our final prototype, decided to utilize cabbage as a source of pigment. And for the prototype, we took a hybrid approach. That means that we took household items and measured them and created 3D printed adapters for them. Inside the fan is a refillable marker with a huge tank. Around it are two syringes holding the solutions. These have bent needles that insert into the tanks. This is all held together with a snap lock mechanism. Around it is an ergonomic holder that has holes. The holes give it a more organic look, but more importantly, we use less materials printing it. On this slide, you can see how it all comes together. And here you can check out the 360 view. And for good measure, here's a close-up. One important aspect that we knew we couldn't skip is sharing. Especially in biodesign, a lot of cool companies develop fascinating methods to create something and then keep it all to themselves for profit. That's why from the beginning, we decided to keep all our learnings and give it back to the community. So if you check out this QR code, you can download all the 3D models that we are using currently, and we plan on updating these files and descriptions on how to recreate our project. As of now, we've tested pHen on textile and paper, but hopefully once the community gets their hands on it, a lot of creative inputs get to us, and we can adapt the pHen to it. As you can see, the different styles can be achieved, like sketches and watercolor. One interesting future application would be an adapter for a CNC machine or a 3D printer. And we'd like to thank the people who made this project possible, Uni, Fetsi, and Malu. Hopefully you got the chance to try it out at the exhibition, and we hope you liked it, and feel free to let us know what you think. Thank you.
Your prototype for the pHen, which was beautifully designed, it seemed as though it had two chambers, one a syringe. Can you explain how that worked? I didn't quite understand how it needed two different chambers of liquid.
Yeah, one chamber, sorry. Okay, one chamber holds the acidic and the other is the basic. For that, we use baking soda and citric acid.
I had a question about the design as well. It seems a lot thicker in circumference and bigger than a conventional pen. I was wondering if that was an aesthetic decision, or was it to have room to house the interior, and how that might have impacted user testing?
It was a lot of factors together. We have looked at a lot of pen designs. In the future, we want to make it even more ergonomic. So we would cut down from the sides in a manner. But right now, because of the nature of the project that we really wanted to get feedback as fast as possible, we decided to go with a simpler approach.
Time for one more? Yeah, Russ.
How does refilling work, if it's possible?
So as for the pens, which contains the pigments, they are refillable, and you can buy them in a store. You just have to twist the top down, and you can basically refill it and twist it back. As for the base and the acid, we have a water mixture with the acid and the base, and with the syringes, you just have to suck it up and put it back to its place.
Thanks, y'all. Well done. Next up, my script just jumped, so I don't know. Give me 1 second. We have Rye Country Day School, Polynation. Let's go, Leo.
Hello, everyone. Thank you all so much for being here. My name is Leo Roth, and I'm a rising senior at Rye Country Day School in New York.
And my project today is called Polynation. I'd like to start off by addressing the problem, or rather the myriad of interconnected problems that subtly define our modern world. In the US, bees pollinate over 80% of all flowering plants. And since 1962, there's been a 90% decline in bees per hectare. This is due to a number of problems such as poor nutrition, habitat loss, urbanization, and the lack of green space in urban areas. The core problem, though, is that bees are a keystone species. Humans, pollinators, flora, and fauna are all interlinked, and when one node in an ecosystem is threatened, the entire network is put at risk. Additionally, the plight of city pollinators is worsened by inefficient and overly anthropocentric human design. In urban areas, bees only forage in a radius of about 0.3 to 1 miles. For reference, that's about 6 to 20 city blocks. Where we're standing today at MoMA is about 2 miles from Parsons. If you think about the amount of green space in nature in that distance, there's not much. So bees are struggling to find food in urban areas, essentially. In creating the concrete jungle and building the metropolis, we've created a hostile landscape for all non-human agents. Additionally, pollination seeks to solve many anthropogenic problems as well, such as environmental justice, urban decay, a lack of green space, and linear design processes. In urban areas, the lack of access to nature can often be a detriment to mental and physical health for humans. This can be an environmental injustice issue because those communities that face urban decay are often minority and or low income. My solution to all this is the polyflower. Designed as an intervention into an unsustainable system, the polyflower is a novel mechanism for the rewilding of urban environments. It's a synthetic flower that is made from an algae-based biopolymer meant to rewild urban systems while literally sowing the seeds for a greener future. There are 3 components to the polyflower. First is sea pollen, a nutritious substitute for bee pollen. The sea pollen is made from citrus food waste as well as certain microalgae, such as spirulina. This provides an immediate life support to urban pollinators that face poor nutrition in cities. The citrus food waste is made from mainly biomass because over 50% of all citrus biomass is wasted during the food production processes of citrus. So the sugary chemical inside the algae as well as the citrus provides all the necessary amino acids, lipids, and carbohydrates for the pollinators that other artificial substitutes just cannot. The second component is the algae-based biopolymer of the polyflower itself. The flower is embedded into the ground, and from there, the flower will begin to biodegrade. And the next step is the native seeds are embedded in the stem of the polyflower so that as the flower biodegrades, it fertilizes the soil, and the seeds will eventually grow into an actual native plant. The polyflower seeks to use regenerative design processes to transform decay into growth across scales. On the product scale, as the polyflower biodegrades, it induces growth for the native seeds embedded in the stem and induces a native ecosystem to become recolonized. On a community scale, pollination uses socio-ecological design strategies to transform communities affected by urban blight into sustainable areas that live in connection with nature. The life cycle of a pollination is based on a new model of ecological succession focused on bioremediation, one in which humans aid in the rewilding of abandoned ecosystems. This all begins when a polyflower is embedded into the soil, bringing new life to a desolate landscape. Next, the polyflower will become a keystone species, as the sea pollen provides necessary nutrients for the urban pollinators that otherwise are starving. Soon, the embedded seeds will begin to grow, providing sustainable biomass for the community and further encouraging ecosystem growth. Lastly, ruderal plant species will quickly recolonize the landscape, transforming the entire ecosystem. As succession progresses, biodiversity will strengthen, offering the community new resilience in face of climate change. Here are some additional renderings I made of a polyflower from a bird's eye perspective and an orthographic one. In designing the flower, I merged the natural and the digital, using parametric design processes to create new topologies, because in this case, form is function, as the design had to be aesthetically attractive for all stakeholders, both human and non-human alike. Additionally, I created a 3D printed prototype made out of PLA or polylactic acid, which helped inform my decisions as I was designing, as I went through the iterative design process using 3D software called Blender. During the production of a polyflower, it's important for me to consider the entire life cycle, from production to consumption to regeneration. During the production phase, I mapped out all the core components and ingredients because it requires a series of materials and biofabrication techniques. The main component of a flower itself is an algae-based biopolymer made from microalgae, as well as bacterial cellulose, organic colorants derived from citrus food waste, as well as calcium carbonate derived from oyster shell waste. The same way that we've created artificial reefs for our ocean, the polyflower essentially attempts to create terrestrial reefs for landscapes above ground. As the product was used, it was helpful to create a roadmap for consumption, from purchase to use. This allowed me to map out all the possibilities of usage, especially in case it gets into the wrong hands. The great thing is there are no wrong hands, because all the user has to do is simply embed the polyflower into the soil and let nature run its course. The most important component of a polyflower, however, is its second life post-consumption. After being embedded into the soil, it will biodegrade, and the native seeds will be able to grow in its place, as the nutrients from the algae will fertilize the soil. I tried to focus on going beyond sustainable design methodologies, instead of trying to reduce environmental costs, creating an entirely net positive solution that focuses on regeneration. As I pursued further research, I was able to begin prototyping and creating biomaterials for the product. First, I created a simple alginate bioplastic made from sodium alginate, glycerol, calcium chloride, and water. And this gave me access to new forms and experimentations, essentially accessing a new material ecology. I was designing in 4D with growth as a new dimension. Additionally, I was able to create a prototype of the sea pollen material using a combination of bio-based ingredients. This allowed me to visualize all the components united with the 3D printed corpus, as well as the orange, sodium alginate, coconut oil, and glucose. The use of biomass from citrus food waste proved to be especially useful because of some hidden properties. For one, oranges have long been used in bioremediation projects, as seen in a case study from Costa Rica, in which a fruit company essentially unloaded 12,000 tons of orange peels onto a landscape, and an entire forest regenerated there on its own. Additionally, within the orange peels, there's chemicals such as E-citrol and Z-citrol, which are chemically identical to the pheromones that a bee will release during foraging when signaling to other bees that it has found food. Also, the chemical D-limonene in orange peels is a repellent of mosquitoes, which is an added benefit. In my design, I was able to base my research on very recent biodesign projects for precedents, such as algal 3D printing from EcoLogic and from Adler Luma in France, which is on display in the moment here, as well as interspecies architectures from Terraform One over in Brooklyn. I'm just a high school student, but seeing all these projects made me realize that collaborating with nature for interspecies change is the way to go. In terms of ethics, the goal of a polyflower is to design with life for life. I have some values on the screen, but overall, this approach has allowed me to design across scales, from product to ecosystem, synthetic biology to circular bioeconomy. Polynation explores interactions and cultivates connections between humans, natures, and technologies in order to create a healthier and more equitable future for all. This has allowed me for interspecies collaboration where there's no one singular hero organism, but rather a grand mutualistic symbiosis, where each stakeholder, each species, helps one another thrive. Here are some of my sources, and I'd like to thank my mentors, Kevin Ferrant, Ella King, and Catherine Bischoff, all my teachers and mentors in this process. They've been amazing. Thank you. Any questions?
Judges. Oh, Corinne. Mike, are you gonna run it?
Wonderful project, thank you. How did the perspective of the stakeholder of bees and insects inform the parametric design that you came up with in the end?
Sure. So as I was designing, I tried to learn more about what attracts bees to flowers. That's actually one of the first things that got me interested in this project. So I tried to create first a model with high surface area, which would allow for increased landing space for pollinators, as well as the pink color, which is used from the bedonins, which is derived from blueberry food waste, is part of the UV spectrum that bees can see because they're highly attracted to bright colors. In my original rendering prototype, it was just white due to my limited materials, but as I would progress, I would begin doing more experimentation with form to attract bees higher. Additionally, the conic and almost runway-like shape of the pattern from the flower petals helps attract the bees as well.
I would like to buy this. Do I just stick it in my garden? Like, can I have this today? And so how do you see in terms of like how people integrate it into the gardens that they have now?
Yeah, so I think there are many routes this can go. I would like to focus on community-based and very local place-based design strategies. So while this is definitely a consumer product and it could totally be used in suburban situations to rewild gardens and such, it's mainly going to be focused initially, at least, on rewilding urban areas that face problems such as urban decay, brownfields, areas like that where bioremediation is a very urgent, necessary problem, and then it would later be applicable to all consumers through local businesses.
So I could have this in like my community garden.
Yeah,
today.
that'd be an ideal situation. Yeah.
Awesome, thanks.
How long would it take for the flower structure to degrade, decompose, whatever the right word is there, and then the native seeds to grow?
Yeah, so it would take about 6 months. The goal of this was essentially that pollen provides a very immediate and urgent solution to the poor nutrition problem because even in winter especially, bees and other urban pollinators don't have access to flowers and pollen because they have to rely on their storage. So if this was planted in the fall per se, then the flower would last all winter, and then as spring comes, the actual flowers would grow in its place.
Yeah, I want this project too, but I don't have a garden. Maybe a windowsill. When you were thinking about rewilding places of urban blight, it wasn't in the presentation, but were you also thinking about city officials or residents of communities and asking them? I mean, I can't imagine why you wouldn't want that, but I don't really know. So did you think about those issues, and would you be thinking about those in the future?
Yeah, it'd definitely be a very, when it comes to rewilding the urban areas, I'd focus on working with the communities to identify their needs, identifying which areas in specific need the most focus for bioremediation purposes, but it'd all be very local and the design would be adaptable. This is just a very basic prototype, mainly modeled after the Germanium maculatum, which is a New York City native flower, but it would be dependent on the communities and I'd be working with them to design for their unique problems.
Awesome. Thank you, Leo. Wonderful job.
Thank you. Thank you.
We're going to go on to our last project, McGill University, Kelserra.
All right, hello everyone. We're McGill University and our project is called Kelserra, an algae-inspired adhesive for use in minimally invasive surgery. So our team is a student-led engineering design team from McGill University in Canada, and here's a picture of our entire team of 23 members. So the context of our project is in minimally invasive surgery, and a large category of these surgeries are related to gynecology. For example, they're used to diagnose and treat endometriosis, which affects 10% of reproductive age women globally. And another example of its use is to investigate and remove ovarian cysts. and it's estimated that 5 to 10% of women in the US will require this surgery in their lifetime. So we examined the current technologies in incision closure, and it's clear that sutures have many shortcomings. Although they have a high tensile strength, they damage tissues and they're time-consuming to apply, and this becomes even more complicated in difficult to access areas of the body, such as those commonly encountered in minimally invasive surgery. Synthetic glues solve all these problems, but they're toxic to internal tissues, so they can only be used on external skin. And finally, bioadhesives have the advantage of being biocompatible, so they can be used internally, but they lack strength, which limits their usability because surgeons will only use a product that is very strong. So the critical question that our project aims to answer is, can we create a bioadhesive that is strong and biocompatible to improve surgical outcomes for women and everyone else by proxy? So to answer this question, we created Kelserra, which is a biomimetic algae-derived glue, and our lab tests show that it's stronger than our benchmark glue, and I have a sample of it up here. But before we show you all the scientific data, let me tell you our origin story. So the story starts deep in the ocean, where algae use polyphenol compounds to stick to wet surfaces and grow towards the sun. One day, a scientist named Rhonda Bitton and her colleagues noticed this phenomenon, and they adapted the mechanism into a surgical glue for wet internal tissues. They showed that it was as strong as the current strongest bioglue on the market, indicating this idea's feasibility. Their project spun off into a startup company, so all the data went private, and we decided to take the same inspiration from algae and develop our own glue in our own direction. To aid in our design process, our medical outreach subteam interviewed experts in the field from the pharmaceutical industry, from academia, and practicing clinicians who are the end users of the product. We learned that the main value proposition of this glue is its ease of use, strength, and speed, as shorter surgery time is critical to ensure patient recovery. Strength is also key because it's very important for surgeons to have a strong product, so the focus of our design was on strength to overcome the main limitation of bioadhesives and differentiate ourselves from other products. We were also advised to consider the regulatory approval process and affordability of the product. And one expert even suggested that we add a color indicator to tell the surgeon when the glue is done curing, and we turned this idea into a reality.
So once we established our concept, we started designing our glue by first creating different formulations and then testing their adhesive strength. So we iterated between formulation creation and strength testing, where the new formulations were based off data from previous strength tests. We determined our strongest formulation so far and tested its biocompatibility. And we're currently at a step where we would still need to do a few more iterations before we move on to clinical trials, regulatory approval, or market launch. In terms of formulation, our glue is mainly composed of 5 ingredients. We have alginate, the polymer that's derived from algae, fluoroglucinol, a compound that mimics algae's adhesion in the ocean, calcium, xanthan gum, a common food thickener, and a curing agent. The strength of the alginate matrix is due to the cross-linking of the alginate strands with the calcium ions, as shown here. And once we add the curing agent into our mixture, the liquid jellifies, and that's when it can be used as a glue. So we created 12 initial formulas and then 3 optimized formulas based off the data from the first round of strength testing. We also added a color indicator based on pH change, and we were mindful to source our products to align with our budgetary constraints. After the formulas were prepared, they were sent to validation for strength testing and cytotoxicity testing. So we tested our glue on pork skin, which is a common substrate used by many other bioadhesive researchers, and we used what's called the lap shear test, where overlapping pork skin samples are glued together, pulled apart, and the strength at which they break is measured using a digital force gauge. We ran into like a major challenge, which was that we didn't have access to a professional testing instrument, so instead we created our own testing apparatus as shown here. We collected the strength test data and found our strongest formulation. Where we could have compared our glue to many other commercial adhesives, such as fibrin sealants, these were very expensive and difficult for us to obtain. So instead, we compared to egg albumin adhesive, which is made by drying egg white, rehydrating the powder, and it has been shown by other researchers to be a viable bioadhesive. And when we compare our glue to the egg albumin adhesive, our glue proved to be much stronger.
So here's the final composition of our formulation, which is mainly made of alginate. And this is what's used for biocompatibility testing.
Cytotoxicity testing consists of measuring cell death after exposure to Kelserra using a dye named trypan blue. Dead cells are porous, meaning that the dye is absorbed through the cell membrane and stains the nucleus a dark blue, which is visible under the microscope. Live cells, however, have intact membranes and thus remain clear. And with this technique, we can easily identify live versus dead cells and derive a cell count. Here are assays under the microscope. We used human bronchial epithelial cells for our trials and conducted 2 controls shown in the second and third panels. Examples of live and dead cells are also circled. We then tested both our benchmark adhesive and our ideal Kelserra formulation. From this data, we derived a cell viability count. Although Kelserra has a slightly lower viability than our positive control, it also has a higher viability than egg albumin adhesive, our benchmark. Overall, this experimentation showed encouraging results, but not without a few obstacles. The human cells, which are extremely sensitive to their environment, pose a logistical challenge in their transportation in between lab spaces. Thankfully, our cell culture survived and proved to be viable. Next, let's take a moment to discuss the implications of Kelserra, both within an ethical and environmental framework.
Kelserra is biocompatible with our living tissue, thus it ensures the safety of the patient. By avoiding sutures, the use of Kelserra results in a shorter endometriosis surgery time. The alginate prevents the risk of surgical complications, infection, blood loss, and reoccurrence of endometriosis for the patient, who will also recover faster after surgery. Plus, faster surgery means less stress and less fatigue for the doctor, which is always good. As I mentioned, Kelserra offers crucial improvements to surgery for both physicians and for patients. By using low-cost ingredients and simplifying the creation process, we aim to make Kelserra accessible to people of all socioeconomic statuses. While we have focused on a specific surgery, Kelserra has the potential to benefit many more minimally invasive surgeries.
One of Kelserra's biggest strengths is its biomimicry of brown algae. Due to this design mechanism, all components of our bioadhesive are biocompatible, biodegradable, and eco-friendly. Brown algae is harvested in underwater farms, employing resource-friendly methods such as sun drying. It is also resistant to ocean acidification and provides essential habitats for symbiosis. Furthermore, brown algae's rapid growth rate of up to 50 centimeters daily makes it a fully naturally renewable resource. Alternatively, alginate can also be cultivated in vitro with bacteria, which is an extremely efficient and resource-friendly method. Perhaps most importantly, brown algae is a key photosynthesizer capable of removing up to 0.55 gigatons of carbon dioxide annually. That's more than the Amazon rainforest.
Although our product seems promising, we did have some difficulties with our testing that we hope to improve as we continue our work with Kelserra. The glue currently exhibits a lower tensile strength than the literature values of similar products due to cheaper glue components. In the future, we aim to address this issue by enhancing ingredient purification and incorporating natural side ingredients that would enhance the adhesion while also keeping costs low. Given more time, we would have conducting more testing, which we will do next year. While our in vitro testing showed promising biocompatibility, further in vivo testing is required in order to ensure safety to human health. We recognize the possibility of Kelserra being used to financially exploit those in medically vulnerable positions. To mitigate this risk, we are committed to pricing our product affordably. We aim to avoid monopolized control of Kelserra by large companies in order to steer clear of single corporations controlling medical products. Here are our references and our acknowledgements. Thank you very much for listening.
Judges.
Could you talk a little bit about how this would be used in practice? Is it presented as a two-component and the surgeon is then mixing in place, given the sort of cross-linking thing?
Yes, so the idea is that it comes in like a liquid form, and a similar adhesive that was developed by other research groups has been shown that it can retain its strength for 8 weeks. So basically you can store it up to 8 weeks and it will still be strong afterwards. So you combine the base liquid with a curing agent, and that's what gelifies it and makes it actually sticky. So it is a two-component mixture. The surgeon would like open the bottle, take out the liquid, and then put in the curing agent, and then apply it to the patient, while simultaneously checking the color indicator in a dish on the side, aided by an assistant probably, to see when the glue is done curing, and then they will know that the strength is good and that that procedure is finished.
Judges.
Hi. I'm curious about a little bit more about the testing. You said that you did test. Forgive my ignorance, but is there any allergic reaction to this? Is that possible? And could you speak a little bit more about the testing that you have done and that you will do?
Yes, thank you for your question. So cytotoxicity testing is actually the first step to kind of measuring potential insults to the bodily fluids and the human body in general. There are much more tests to be done, such as immunogenicity and allergic reactions. Cytotoxicity testing just gives a general overview of the initial insult to the cell. So does it kill cells? And we chose human bronchial epithelial cells for this because in research, they are known to be very germeric cells and are also easily put in suspension, so for liquid testing. So we have yet to test for market-based and market approval tests in these assays. Our issue is these types of assays, since they're standardized, are extremely expensive and monopolized, and so we do require more time and more fundraising to be able to do this.
Thank you for the wonderful presentation. I just had a question. I'm hoping that you could speak a little bit to the need for this particular glue over other biocompatible glues that are already on the market.
So the main limitation, as I mentioned, of bioglues currently is that they're not strong enough, and a really important factor for surgeons, which we learned through our interview process with them, is that they really value strength. And their main worry when we introduced this concept to them was like, well, what if it breaks apart? Like, how do I know it's actually going to stick? So that's like really the main concern for surgeons. And this is what bioadhesives lack. So that's why with this glue, as we've shown that it's stronger than our benchmark, and as we mentioned, we need to do more testing with the commercially available benchmarks, which are ridiculously expensive and so out of our price range. But by designing for us better strength, we would overcome that limitation that the whole category of bioadhesives suffers from, and could maybe make internal bioglues like more used by surgeons who really value the strength.
Awesome. Okay, that's it. Wonderful job. Thank you, McGill. Give us a second to transition to the podium. Hello. Okay. Awesome. All right, everybody. We are at that time of day where we're going to go to lunch, but wait for a second so we can explain things. Maybe take your phones out really quickly. So we have about an hour and a half of lunch. We cannot eat on the premises, so we have a map here for you all of public places all throughout. There are parks here. There are like public spaces that you can go and sit outside or inside, so you can kind of go throughout the city and see and they're very close to MoMA, so just keep track of time. Judges, please stay here with us. We have directions for you, so don't leave for lunch just yet. And then, yeah, that's it for me.
So I wanted to mention a couple things. We have some lunch excursions. This is a little bit off because we were running a bit behind, but if you sign up for this Swarovski excursion, you should have gotten an email about the access with like a QR code that explains the process. We'll also be meeting here in the lobby where lunch is served at 1:10 p.m., not 12:45. just in case anyone wants to walk over together. But if not, you can meet at the offices at 10 East 53rd Street, which is just down the block. The presentation will start at 1:20.
And this is if you registered ahead of time.
Yeah, this is for people who registered already for this Swarovski ski excursion. So if you don't know what I'm talking about, you can ignore everything I just said. And then also, if you would like to explore the MoMA galleries during the break, this is something that MoMA is graciously offering. We'll have tickets available for pickup in the lobby when you return from lunch, because we can't have people eating on the premises. So once you have lunch and come back, there will be tickets available in case you want to explore MoMA during the lunch break. And please be sure you bring back your badge in order to get re-access to MoMA later today. And so yeah, for those of you joining us online, feel free to stick around. We'll be broadcasting over an hour's worth of videos from artists and designers in our community. And everyone, please be back by 2:20 p.m. because we have a performance by artist, performer, educator, and technologist Smita Sen before the awards ceremony. So we'll see you all back here at 2:20.
Thank you. Okay, cool. Judges, we're just going to wait till the room clears out and then we'll talk.
Implantable into the body. And if we're going to be implanting back into the body, what does that mean? How would we redesign our bodies, and what would we make ourselves into? As an artist and designer, it's also really interesting to consider what would we make if we no longer need to use inanimate objects like metal and fibers. If we can now use the building blocks of life, actual cells and tissue, what would we make?
This is Martha and George, who are soon to be mother and father. They made the choice to fulfill their pregnancy with parturients. Parturients, which provides artificial wombs for anyone for the period of 9 months. This is the parturient's pod. The baby is nurtured through this object. It is similar to a pregnancy, but without the physical restrictions. Throughout this period, the family can live and work as they would normally do, whilst also being able to bond with their child. George A. Award, 29, is a journalist who is engaged with several interesting projects, due 2051. This is a communication device, which is connected with the parturient's pod. The device can record desired moments of the daily life of the family. With its integrated microphone, it can record music or any other sounds desired. This is how the baby gets used to the voices of its future parents. Martha Aylward, 30, CEO of a company she started, works to ensure that the company meets their goals. It is almost time to go home. This is a portable communication device used when not at home. In between her meetings and lunch breaks, she can communicate with the baby. By simply switching it on, you can feel the baby kicking and moving. Carefully touching and rocking the device will send movements to the pod, which the baby can also feel. George is setting the table for dinner. There's Martha, home from work. George has chosen a wine to go with their dinner, and that's all right because Martha isn't carrying a child. Martha is cutting some ingredients for a special recipe that will set the preference of taste for their future child. The most important part of the growth process is the nutrition intake and the future preference of taste for the baby. The pod keeps track on what the baby has eaten and what it needs to stay healthy. After the food is prepared, it is placed in a capsule and placed in the parturient's pod for the baby. At night, the pod goes through the updates made by the hospital with the information obtained during the day. This ensures the baby is healthy and the pod's program works without errors. The day is almost there. This is the final moment in which the pod's lock is lifted and the baby finally meets the family. George, Martha, and newly born Robin, one big happy family. Look how happy they are.
My name is Dr. Jennifer Willett. I'm an artist. I am a professor in the School of Creative Arts here at the University of Windsor. I'm a Canada Research Chair in Art, Science, and Ecology, and I'm the Director of Incubator Art Lab. I'm here to talk to you today about a new project called When Microbes Dream, an exhibition at Kapelica Gallery in Ljubljana, Slovenia in October 2021. Since then, the exhibition has been presented twice in Portugal at Ectopia Experimental Art Lab in Lisbon and I3S at the University of Porto, each involving a different version of the title artwork, When Microbes Dream. When Microbes Dream is an art installation that includes performative human and non-human interactions and hands-on biotechnological protocols. Together, it presents a feminist science fiction where biotechnology manifests interspecies collaboration, reproduction, theater, and storytelling as a means to reimagine our shared biotech future. The exhibition consists of 4 works. The first being the Biosafety Puppet Playhouse, which is a puppet theater that is also a biological safety cabinet. And it is decorated with this sort of Baroque Rococo patterns that are also based on the local ecology here in Windsor, Ontario, Canada. The next piece is called Baroque Biology Paper Theater. This is a series of digital prints that are photographs of GMO bacteria in petri dish environments, laid and displayed on backgrounds of theater settings and designs. They're intended to create this image of the bacteria performing for us, sharing for us their information, their knowledge of biotech processes. Laboratory Ecologies is a complex installation that involves a sort of tent camping aesthetic for a lab that involves large animals, small animals, and microbes all working together in a cacophony towards a biotech research project. There are no humans in this installation. The humans are only the observers, the learners. And the final piece, called When Microbes Dream, the title piece for the exhibition, is a wall mural of microbial ecologies layered with a liquid spiral installation on top, giving this dream world, thinking about what it might be like for the microbes to live and cohabitate with other organisms in our lab. With this project and many others, I perform an alter ego character, the gentleman scientist, the epitome of the colonial traditional scientist who becomes absurd and defunct and self-deprecating as he or she moves through this biotechnological space, sharing with audiences access and discussion to biotech projects. Each artwork presents a complex vision of organisms large and small, mammals, microbes, plants, and insects, working in collaboration to communicate biotech processes to human audiences. This strategy deprioritizes the human scientist as the dominant organism in a robust laboratory ecology, rather presents the perspective of non-human organisms living, reproducing, performing, and dying. Of course, this is an absurdist and impossible task to ever imagine what biotechnology is like from the perspective of our microbial collaborators. It can only ever be a vision of what humans might think that microbes might dream. When Microbes Dream is produced with the support of the University of Windsor, the Incubator Art Lab team members, and the microbes and organisms that we share our lab with.
Contaminated soils, communities saddled with lack of employment. These are the legacies left behind by the decline of the mining industry in South Wales. However, this contaminated soil can now be used for the production of something called a quantum dot, with the help from a very familiar garden friend. So, what exactly is a quantum dot? It is an incredibly small semiconductive particle. It emits different coloured light in different sizes when excited by light or electricity. They have been used in a range of different applications such as biological imaging, improved LED screens and solar cells. The industry is set to be worth over $11 billion by 2026. In 2013, researchers at King's College found that they can be biosynthesized using the earthworm through a process called mutual antagonism. When earthworms eat soils containing metals, they produce enzymes called metallothionein in its gut. The metallothionein grabs hold of the metals and stores it away in special cavities inside the cells. It reacts with another toxic element to make a safer compound. This is the quantum dot. Professor Mark Green of King's College explained,
"Extracting a quantum dot made in a worm is a cheaper, more sustainable process than making quantum dots in the laboratory."
Quantum Industries is one company striving to exploit this phenomenon. Working with a disused coal mine, they have biologically engineered a giant hybrid earthworm optimized for high-yield production of quantum dots. These earthworms are hatched in secure wormeries inside the mine. To attract the wormer runs a light electric current into the soil. The worm is then tickled to release the fluid containing the dots. This mine uses its cadmium and selenium contamination to create a cadmium selenide quantum dot. The BBC interviewed Margaret Howell, CEO of Quantum Industries.
Though we intend to sell quantum dots on as a green and cheaper alternative, our real aim is to generate clean energy using the quantum dots in our solar flags.
Quantum Industries are now fabricating innovative solar flags using spray-on solar cell technology. The flags are raised around the mine, powering itself and the nearby community. Quantum Industries hopes this would re-establish mines as a source of energy, bioremediating the coal industry's pollution and revitalising the industry in the area. Olivia Bargeman, BBC News, Blenheim.
いつか出会い、愛し合う二人は運命の赤い糸で結ばれている。 そんな神話のような糸を紡ぐ蚕が2016年この世に誕生した。アートと科学の創造で新しい神話が生まれる時代始まったのかもしれない。 オキシトシンを作る遺伝子をこの卵に注射すれば恋に落ちる運命の赤い糸生み出せるはず。蚕の目が光れば成功のサインなんだけど。俺ちゃったベイビー好きであってたのに運命のレイディー私海のようにザナビみたいに。幸彦先輩、私ももう大人だしオキシトシンくらいで恋が叶うなんて、恋はそんな簡単な話じゃないだなんて、そりゃ分かってるんですけど、でもそんなことやってみないと分かんないじゃないですか。失敗は成功のもとだって施術教授も言ってたじゃないですか。だから私作っちゃったんです。ねえ、新しい神話一緒に作ろうよ。そうしそう愛ないとは言わないで高鳴る野望は止まんない。ゴーゴーゴーダンなんても言わないで運命なんて結果オーライ。
Rhapsody for an Expanded Biotechnological Apparatus is a tactile sound installation located in the elevator of the Tang Teaching Museum that connects hidden artifacts of science to familiar domains of the everyday. I'm Laura Splan, and I'm an interdisciplinary artist. My work explores the intersections of science, technology, and culture through embodied interactions and sensory engagement. The installation re-envisions the elevator as a biological cell and its visitors as proteins as they're prompted to engage with a haptic textile sculpture while listening to a sonic tour of a biotech laboratory. The soundscape is a wandering journey created from recordings made during my artist residency at the Science Center in Philadelphia. Robotic movements of machines, gurgling dish drains, and conversations among scientists come together to create a soundscape entitled chaperone. In biology, a chaperone is a type of protein that helps other proteins fold properly inside the lumen of a cell. The accompanying textile sculpture, entitled Lumen, prompts visitors to embody the biological process of protein folding when sitting on the rug made from the fiber of laboratory llamas and alpacas who are used to produce antibodies for human drugs, including vaccines. Instructions guide visitors to remove shoes and sit cross-legged as a choreographed gesture that embodies the folding of proteins inside a cell's lumen. Tactile transducers beneath the rug vibrate with the bumps, clicks, and bangs of laboratory machines in the soundscape. The fiber for the sculpture was donated by a vivarium at a laboratory that produces biological products for biotech research and pharmaceutical industry. I hand wash, card, and spin the fiber into yarn for my textile sculptures and installations. The work questions notions of the presence and absence of bodies, evoking the mutability of categories that delineate their status, and renders the elevator as a space for contemplation of the unseen labor of both humans and non-humans within an often invisible system.
Hi there. Welcome to the SBA BioArt Lab, where we've been designing the future of food for over 30 years. Good. This laboratory, the first of its kind, is where biologists and artists work together tackling some of the world's biggest biological concerns. Over the years, we've spawned some unique results, but nothing as successful, impactful, and delicious as this. Meet the Mayo tomato, the amazing protein-packed fruit that started it all, a biological breakthrough that changed the way we think about protein. I have something amazing to show you. Randy, get the thing.
As you wish.
Before the Mayo tomato, humans relied heavily on animals for their daily source of protein. With a growing global population and a strained agricultural system, we recognized animal protein was not an ecologically sustainable source. There had to be an alternative method. we put together some of the brightest scientific and artistic minds we could find, and the result was impressive. Take a look.
This will only hurt a little bit, JK.
In 2016, the BioArt team at the School of Visual Arts engineered the world's first fruit to produce the same protein, myoglobin, found in animals, the myotomato. They found plants to be deficient in megadoses of protein necessary for a nutritional human diet. So they translated a DNA sequence found in beef and inserted it into a tomato's genome. The result? One little tomato possessed the same amount of protein found in an 8-ounce steak. This breakthrough creation caused quite the stir. It seems not everyone was a fan of this amazing scientific achievement.
GMOs are dumb and stupid and poison.
Of course we're not happy.
However, in the year 2030, mad cow disease almost completely wiped out the cattle industry, creating an urgent need for the myotomato's amazing protein power. And soon, hearts, minds, and mouths were open. The myotomato ushered in a global movement that ultimately ended our need for animal consumption. Not bad for a group of art students, if you ask me.
We like to define things and what doesn't, because change means uncertainty, and uncertainty can be daunting. But stopping change is as impossible as stopping time, and focusing on the impossible is a distraction. What if instead of exclusion, we strove for balance? What if instead of eradication, we focused on utility? What if we embraced our environment the way it is, and we kept embracing it as it continued to change? And what if, when we stepped in to help, we did it not to destroy what doesn't belong, but to put what's out of balance to good use? We need to make invasive species material sourcing as commonplace as organic, post-consumer recycled, grass-fed, free-range, biodegradable, free trade, VOC, and hormone-free. It needs to be policy. But first, it needs to be designed. In our region, the Great Lakes region, the introduction and proliferation of zebra and quagga mussels have overwhelmed native species, their habitat, and our infrastructure.
efforts to remove them have been ineffective and in some instances, even more harmful to the environment. As color and material designers, we wondered what might happen if we treat them not as a biological threat, but as an overabundant material. We asked, what are they made of, and what can we make from them? Mussel shells are 95% calcium carbonate. Take them up to 1,000 degrees Celsius, and you get calcium oxide. Calcium oxide, lime, sodium oxide, soda, and silicon dioxide, silica, make glass. As bioaccumulators, zebra mussels filter the water. They filter out pollutants and plankton, minerals and trace metals, some of which give glass its color. A glass derived from zebra and quagga mussels will express the color of their ecosystem. Invasion, to region, to chemistry, to color, to identity, to belonging. We collected the first batch of mussels from Lake St. Clair, where zebra and quagga mussels were first introduced in the 1980s. Hitching a ride on ballast tanks of cargo ships from Russia and Ukraine, we boiled them, cleaned them, ground them up, and sifted them into a coarse powder. We worked out the chemistry, the molar calculations for mass and batch recipes. We designed the kiln program to pause on the way up to control the release of CO2 and H2O as sodium bicarbonate transforms into sodium carbonate, which transforms into soda, and as calcium carbonate transforms into lime. And as we paused on the way down and modulated the rate of cooling to swiftly transition through the vitrification range and to slowly drift through the anneal. It turns out the color of Lake Michigan glass is the color of icebergs and tropical atolls, the color of water. We're told it's a copper blue from the high copper content of Lake Michigan. It's beautiful, and we can't wait to see the color of Lake Superior or Lake Ontario. We can't wait to see each lake, comparing glass from the beach to the lake bottom, taking a biochromatic tour of the water column. We can't wait to rediscover our region through the brilliant blues or greens, or even yellows or rose pinks of what we keep calling an invasive species. And we can't wait to see what we can make out of zebra glass.
trying to find you. Now I'm useless. Used all of me trying to find myself. Now to trade my entire being with the figure behind the screen. Much more than I've ever been. Allowing to my body to accept is easy. Allowing to my body to accept is easy. It's easy. Allowing to my body to accept is easy. It's easy. Allowing to my body to accept is easy. It's easy. Lost all of me trying to find you. Now I'm useless. Used all of me trying to find myself. Now to trade my entire being with the figure that's behind the screen. Much more than I've ever been alone. Allowing to my body to accept is easy. Allowing to my body to accept is easy. It's easy. Allowing to my body to accept is easy. It's easy. Allowing to my body to accept is easy. It's easy.
Back2You is a service that enables its users to regularly sequence their gut microbiome and home, maintain a database of their own sequencing results, and regain their lost bacteria. Back2You brings your bacteria back to you. This is Mary, 26 years old, a resident of New York City. Today is February 24th, 2023. As always, Mary wakes up to her alarm and goes to bathroom. Today, when she enters the bathroom, she activates the Back to You sequencer by saying her name, Mary. After she poops and wipes her butt, she takes the backup pad that comes out of the slot and wipes again for sampling. She folds the pad and puts it back into the intake slot of the sequencer. She then flushes the toilet and starts her morning routines while the sequencer starts sequencing her sample. Later, the sequencer will send the result to Mary's personal Back to You cloud. Mary often sequences her stool sample at home to monitor her health conditions and has built her own gut microbiome database. 2 weeks later, Mary gets a bacterial infection and is prescribed with antibiotics. Over the course of her treatment, she gradually loses part of her bacteria community. At the end of the treatment, Mary opens up the Back to You app on her phone, where there is her personal time machine that has all her past reports. She scrolls back a few times and picks the February 24th report based on the app recommendation. Then she places an order of a 7-day bacterial supplement pills, customized to her gut microbiome condition as it was on February 24th. A few days later, Mary's Back to You order is delivered to her door. Mary starts to take her Back to You pills every day for 7 days. Over the course of taking the Back to You pills, Mary gradually regains her bacteria community. This is how Back to You works.
Invasive species are at our doors, barging in unannounced, causing nearly irreversible damage. Now it's our job to get rid of them, and we start with these. Carp, the bottom feeders. There are 3 main species: big head carp, silver carp, and black carp. They eat 120% of their weight in zooplankton and phytoplankton, a crucial food source of our walleye, bluegill, and smallmouth bass. Therefore, we need to eradicate these species of carp from our waters. Complete annihilation is necessary. These carp fight hard battles. We've already tried multiple ways of eradicating them. Now we are developing a new approach. Using the latest advancements in technology, we have designed a bioweapon to fight against the carp. After years of iteration, we are finally ready to release it to the public.
There is a looming threat to America and the Midwest. 139 invasive species currently inhabit the waterways of Indiana alone, causing a variety of negative ecological impacts. River Defenders arms fish that are native to the White River, such as walleye. With our help, they may have a fighting chance. Combining human advancements in weapons technology and the natural capabilities of aquatic life, this idea proposes that a military-grade tactical vest be attached to native fish. The vest is equipped with 2 items: a waterproof camera that provides a live feed to the River Defender's operator, and a remote-controlled firearm. This firearm follows in the steps of the Heckler & Koch P11, both in its construction and history. The P-11 was one of the many products of weapons development during the Cold War and served as a direct competitor to the Soviet Union's SPP-1, which had been produced 5 years prior in 1971. At the time, frogmen could only engage in hand-to-hand combat with knives. The development of weapons like these expanded the possibilities of underwater combat. The P-11 was designed with the knowledge that traditional bullets don't travel well through water. It features 5 barrels that hold flechettes, or metal darts that allow for better accuracy and range. When fired, the P-11 is electrically ignited from a battery pack within the gun. River Defenders builds upon this design with increased ammunition capacity and remote operation via Bluetooth technology. With an official state-of-the-art app, any Muncie resident can connect their smartphone to a River Defender within a range of 800 feet. Once connected, the user can obtain a fish-eyes view of the White River from the camera attached to each fish. Using the invasive species identification software, the user can fire the weapon when a carp appears, taking an active role in combating ecological imbalance. As bizarre as this idea may seem, the U.S. military is no stranger to creating alliances with wildlife. For example, in World War II, engineers considered using bat bombs as a weapon against Japan. Once napalm was attached to Mexican free-tailed bats, they would be released to roost in buildings and hard-to-reach places. Another example is Project pHen, which utilized the cognitive abilities of pigeons to guide missiles. The pigeons were trained to recognize certain targets and then placed in front of a screen with sensors. The birds would peck at the screen when the target moved away from the center of it, maintaining the bomb's glide path. River Defenders is a testament to the new bonds we may forge with our natural environment in order to protect our traditions and values. In today's society, there is a disconnect between parents and their children due to the fact that most parents were raised without technology. Their children, however, are raised in the digital age and spend increasing amounts of time indoors on phones or tablets. River Defenders bridges this gap between parents and children, creating a new family bonding activity that both generations can enjoy. Over the past 2 decades, the cleanliness of the White River has also become a major cause for concern. As the river gradually becomes more saturated with heavy metals, PCBs, and other pollutants, recreational activities are becoming unsafe. In fact, River Defenders may replace the traditional American pastime of fishing trips. Gone are the days of old-school fishing. Now, with River Defenders, a new, exciting pastime exists. The future is now. The River Defenders must work together in order to form a more perfect union to ensure the peace of the White River. It is your time to take action.
River Defenders has a practical function to engage and mobilize the Muncie populace in protecting the ecology of the White River. It is also a Band-Aid solution that turns human conquest of the natural world into a violent, interactive spectacle. As an example of both biodesign and critical design, River Defenders tackles both environmental and social issues at once. Rooted in the Italian radical design movement of the 60s and 70s, critical design challenges an audience's preconceptions. It's meant to provoke new ways of thinking. Anthony Dunn and Fiona Raby define critical design goals as raising awareness, exposing assumptions, provoking action, sparking debate, and even entertaining. Oftentimes, these projects explore speculative futures, ranging from dystopian to utopian. River Defenders aims to do just that, using the topic of invasive species as a point of departure.
Stuart Hall, the cultural theorist, once said, "There is no understanding Englishness without understanding its imperial and colonial dimensions." The United States has used this idea for centuries to villainize the other. Under the guise of protection, the U.S. then resorts back to its usual methods of complete eradication and declarations of war. Time after time again, these methods prove wasteful and unsuccessful. Using these ideas in the previous video conveys how the saturation of xenophobia further warps any chance we have of enacting reasoned and positive change. If we continue to view every problem through these lenses of fear and hate, our needed solutions will be unreachable.
River Defenders was created to highlight issues with the war on invasives. In the field of ecology, some experts are beginning to take issue with rhetoric surrounding invasive species. The militaristic idea of complete eradication ignores positive qualities that non-native species might bring to an ecosystem. One study showed that, quote, invasive species research uses significantly more militaristic language, end quote, than other subfields of conservation. The power of messaging like this shouldn't be understated. In the short term, rallying communities to declare war on certain species could be helpful. However, it may do more harm than good in the long term, distorting the public's understanding of ecology and reinforcing xenophobic tendencies. This conversation gets more complex as we realize that many of our beloved commonplace species were once considered alien or non-native. While not deemed invasive, the honeybee was imported to North America from Europe in the 17th century. We now depend on this poster child of conservation for billions of dollars worth of crops. However, it could be deemed invasive as soon as it begins outcompeting species of wild native bees. Examples like these show the blurry line between helpful and harmful. We then must add climate change into the equation. Climate change is rapidly redefining relationships between all organisms. Consider the tamarisk shrub, for example. Though it has outcompeted many native trees and uses large amounts of water, it has begun to provide habitats for endangered species like the southwestern willow flycatcher. It is also hardy and drought tolerant, addressing a problem that we will face in the near future. Invasives like these can withstand shifting temperatures and low resources, and may be the only way our ecosystems can survive. Some scientists even predict they could help us preserve a working ecology. The damage caused by invasive species like cane toads and bighead carp is irrefutable, and River Defenders doesn't aim to understate it. However, if we refuse to explore new roles they might play in this ever-changing environment, we will keep falling back into costly, unsuccessful methods time after time again. Let's work to build new solutions that look toward the future.
Open eyes, ears, get ready to feel, taste and smell the landscapes of your inner cellular sensorium, a flexing and porous space, alive with possibilities. Within this world, our living cellular kin, Olks, are guardians of the 5 human senses, and each connected to a kingdom of life. In this meeting between our sensory selves and the inner worlds of our cellular beings, futures unfold. Now they invite you to journey along into their cellular habitats, your body, to begin a sensory cellular ceremony. Enter one of the portals. Look at the screen. Focus on the orc in the center, guardian of your human cellular sight sensorium, connecting you to other bodies across the animal kingdom. The image you see is being relayed to you through your soul windows. Watery gelatinous orbs, crystal balls, your eyes. The image you see travels through layers of cellular strata, through a film of watery gelatinous cornea, epithelial cells, keratocytes, and endothelial cells, past the colorful facade of your iris, a lacy meshwork of fibers and pigment granules, down the black hole of your pupil, a portal into a crystalline lens, refracting the image, focusing it into a sharp form before it flows through a jelly-like pool, arriving at your retina, a thick forest of cells with long tentacular arms, pigment cells, rods,
cones, horizontal cells like polar cells, reaching out to one another, holding on to one another to form a network, a community which can uphold your vision.
Live from New York, where the world's first Cas9 opened earlier today despite ongoing protests. The public backlash first started in 2051 when the Universal Genetic Screening Law was passed, requiring preventable genetic disorders to be removed from embryos in order for them to qualify for universal free healthcare. Since then, scientists and doctors have been inching closer to creating fully designed humans, what the public refers to as designer babies. The law states that parents are not allowed to handpick desired genes for their offspring, but Cas9 puts chance back into the equation. We spoke earlier to Cas9's first family unit to hear a little bit more about how it works.
All of our genes are in our 23 pairs of chromosomes. At the Cas9, our chromosomes will be copied and stored in the gene bank so they can later be recombined and synthesized to form the genome of a new baby. We play around with bébé roulette for each chromosome. The wheel ensures a randomized selection, but players can increase their odds by betting extra chips.
And what if you don't like a certain gene on a chromosome assigned to your baby?
CRISPR tokens, just CRISPR them right out.
This is Brianna, one of our parents playing at the Cas9 tonight.
I'm Brianna, daughter of the president and CEO of the gene bank in the Cas9. I'm paying first to have this baby, and I feel so blessed that the government has given us access to this sort of technology. It's a really great option for people like me. Yes, it's expensive, not everyone can afford it, but what I can afford is to have a baby that's just average.
I feel lucky to be chosen to be part of this group. I grew up before humans were screened for genetic diseases, and I've had really crippling medical bills because of it, so I know I can't afford my own baby. But Brianna hired me as the primary caretaker and genetic parent of our baby because I have blue eyes, so I guess I got lucky after all.
This is truly an amazing step in the scientific community. Genetic regulations have hindered mankind to push the limits of the human race, but Cas9 are a step in the right direction. Why should we keep settling for natural selection when we can select whatever we want?
Now let's check in to see how the game is going.
Alrighty, chromosome 9. Chromosome 9 may determine your child's eyebrow thickness, forehead freckles, lip width, blood type, and vulnerability to malignant melanoma. Place your bets.
Okay, the girls are placing their bets. Looks like Brianna's got a lot of chips on the board. Aaron has next to nothing. Now they're spinning the wheel. And Molly wins!
Oh no, not those eyebrows on my baby. CRISPR those out right now.
What? Are you really going to risk editing my chromosome? What if it messes something up? It doesn't work every time, you know.
You could replace your eyebrows with mine.
Oh, please.
How dare you?
Whatever. I still won the chromosome, and I choose Aaron's chromosome number 9 to complete the pair. Next we'll play for chromosome 17.
Looks like the karyotype is shaping up. We'll check back in with them a little later.
Now let's hit the streets to hear from some of the protesters outside the Cas9. Protesters are gathering to voice their outrage towards what some are calling the next eugenics movement.
This Cas9 needs to be shut down now before we raise an entire generation of elitist superhumans. Our government should promote equality, but this government-run Cas9 is promoting rampant inequality.
Some say they fear being further stripped of their reproductive rights. Others are comparing this to the 2020 reversal of Roe v. Wade.
Cas9 are mining all of our genetic information and selling it to the highest bidder. They say they want to maintain diversity, make sure our kids are healthy, but they just want to control procreation. They want to decide who gets to be a human and who doesn't. Cas9 are promoting eugenics. They don't want impurities. They don't want my imperfections. It's ridiculous. It's ridiculous.
In a world where plastic rules all, 90% of toys are made of some sort of unrecyclable plastic.
Our brave biomaterial rebels, Fungus and Scooby, have come to save our planet. Together, they will battle the evil forces of plastic. But wait, they can't do it alone.
Join their mission of ridding the world of plastic, one BioBuddies kit at a time.
We're the BioBuddies!
Our mission is to provide you with eco-friendly toy kits to explore, play, grow, and create with biomaterials. As the world is becoming more aware of environmental sustainability, it's time to move away from plastic. This is where biomaterial design comes in to save the day. Our biomaterial growing kit incorporates science, nature, and art to make biodesign more approachable. We've designed 2 biomaterial growing kits, Scooby and Fungus. Scooby is a kombucha growing kit, and Fungus is a mycelium kit. Each kit includes the necessary supplies to grow the biomaterial and explore with it. The average toy fad lasts 8 months. Our goal is to create a sustainable kit that can be played with, explored, and developed to sustain interest over a long period of time. The entire exploration journey cycle is accessible, educational, and fun.
We tap into the waste stream by feeding our cultures with used coffee grounds, sugary drinks, and tea leaves. First, users grow their biomaterials.
Then they can play and explore through personalized projects with their materials. Of course, not all projects will succeed, so we encourage users to celebrate their fails.
In the future, we hope that we can sell our kits online or even in retail stores like Target to people of all ages. As our kits don't fit into a specific category, we think that they would best fit into the craft or miscellaneous kit aisle.
Help us save the earth by redesigning the toy industry.
BioBuddies out!
Can artists and designers working with technology collaborate with living organisms in a way that is meaningful and mutually beneficial? And can this collaboration act as an essential demonstration of how our electronic-dependent society can change the way we consume and use personal electronics like phones and computers? We formed the artist collective SlimeVolt to determine exactly that. SlimeVolt is an artist-led collaborative that aims to disrupt the modern technology paradigm by incorporating biological components within existing electronic systems, not simply as a means to replace computer parts, but rather to foster a new kind of caring relationship with our electronics. The current culture around our consumption of electronics is entirely unsustainable. The almost immediate obsolescence of personal devices has led to an annual accumulation of 50 million tons of e-waste alone. Rather than simply providing new biological materials to replace electronic components that in turn would become a new kind of e-waste, our collaborative decided to address the cultural problems around overconsumption itself. As a group, we created an instrument that can act as a model for creating new kinds of symbiotic relationships with our personal electronics. We chose to collaborate with the organism Physarum polycephalum, a user-friendly protoplasmic slime mold used commonly in laboratory settings. Physarum polycephalum is a dynamic and fascinating organism that's unique abilities and physical intelligence has continued to resist classification and challenge the greater scientific community's understanding of intelligence. Physarum polycephalum has the ability to solve basic mazes and has impressive pathfinding capabilities. It also is able to respond and to thrive in states where it's exposed to electrical stimuli, making it a perfect fit for our foray into biocomputing.
Originally, we were introduced into biocomputing through the work of Eduardo Miranda, the Director for Computer Music Research at the University of Plymouth, as well as the work of Andrew Adamaski, who runs the Unconventional Computing Lab at the University of West England in Bristol. Adamaski, in particular, has been researching slime mold for over 10 years, and in his text, Physarum Machines, provides us with an experiment that allows us to measure slime mold's electrical potential. In this setup, we would place conductive tape on two ends of a petri dish filled with agar. By attaching slime mold's favorite food on either end, which are rolled oats, slime mold would grow in between the two parts and create an electrical bridge. We would then measure this connection as a resistance value. We found through this experiment that as slime mold grew, its resistance would increase proportional to its growth. We then took our setup into what's known as an oscillator circuit, which takes an electrical current and turns it into a square wave. So this is plugged in. That's so cool. This is our slime sound. This design is at the core of so many early electronic synthesizers and costs only a few dollars to create. In our circuit, we were using a 3-volt battery, which is a size commonly seen in hearing aids, and outputs a safe voltage that should have no effect on our cultures. As a group, we were able to create over 50 plates and found no harm in the organism in any of our experiments. We were then able to create personalized setups to play with and perform with each other. Turning slime mold cultures into an 8-step sequencer, or plugging it into an old toy and hacking how it used to sound. We then shared our progress in class critiques and got feedback from students and faculty, as well as artists and musicians around Chicago giving us some of their best advice. Through this experimentation, we became fascinated by the care relationship that began to develop between ourselves and the organism. This began to fuel our philosophy on collaborating with living components in electronics. To begin to put these ideas to test, we narrowed our scope towards DIY electronics communities, making our practice accessible to them by designing with readily available devices and sharing our entire protocol on existing platforms, as well as on our website with instructional videos on how to get started. In our latest model, we designed a terrarium for our slime molds to live in. We've attached our analog circuit that we mentioned previously alongside with the new integration of a Raspberry Pi. This has allowed us to add in a functionality for time-lapse recording and measure in data such as temperature and other parameters to further push our care element. With an overall goal in providing an open source and customizable setup to care for and record information from slime mold. As a collective, we believe that by introducing individuals on how to care for living organisms within existing machines, that we can foster a new kind of relationship with our electronic devices that encourages collaborations with the living world.
To me, there's so much awe and mystery in the way that life is formed. It's an intelligence beyond our understanding for all these parts to come together in perfect union. I created this as a piece of artwork, but in the future, we could potentially use this same method to make bone grafts that are implantable into the body. And if we're going to be implanting back into the body, what does that mean? How would we redesign our bodies, and what would we make ourselves into? As an artist and designer, it's also really interesting to consider what would we make if we no longer need to use inanimate objects like metal and fibers. If we can now use the building blocks of life, actual cells and tissue, what would we make?
This is Martha and George, who are soon to be mother and father. They made the choice to fulfill their pregnancy with parturients. Parturients, which provides artificial wombs for anyone for the period of 9 months. This is the parturient's pod. The baby is nurtured through this object. It is similar to a pregnancy, but without the physical restrictions. Throughout this period, the family can live and work as they would normally do, whilst also being able to bond with their child. George A. Ward, 29, is a journalist who is engaged with several interesting projects, due 2051. This is a communication device, which is connected with the parturient's pod. The device can record desired moments of the daily life of the family. With its integrated microphone, it can record music or any other sounds desired. This is how the baby gets used to the voices of its future parents. Martha Aylward, 30, CEO of a company she started, works to ensure that the company meets their goals. It is almost time to go home. This is a portable communication device used when not at home. In between her meetings and lunch breaks, she can communicate with the baby. By simply switching it on, you can feel the baby kicking and moving. Carefully touching and rocking the device will send movements to the pod, which the baby can also feel. George is setting the table for dinner. There's Martha, home from work. George has chosen a wine to go with their dinner, and that's alright because Martha isn't carrying a child. Martha is cutting some ingredients for a special recipe that will set the preference of taste for their future child. The most important part of the growth process is the nutrition intake and the future preference of taste for the baby. The pod keeps track on what the baby has eaten and what it needs to stay healthy. After the food is prepared, it is placed in a capsule and placed in the parturient's pod for the baby. At night, The pod goes through the updates made by the hospital with the information obtained during the day. This ensures the baby is healthy and the pod's program works without errors. The day is almost there. This is the final moment in which the pod's lock is lifted and the baby finally meets the family. George, Martha, and newly born Robin, one big happy family. Look how happy they are.
My name is Dr. Jennifer Willett. I'm an artist. I am a professor in the School of Creative Arts here at the University of Windsor. I'm a Canada Research Chair in Art, Science, and Ecology, and I'm the Director of Incubator Art Lab. I'm here to talk to you today about a new project called When Microbes Dream, an exhibition at Kapelica Gallery in Ljubljana, Slovenia in October 2021. Since then, the exhibition has been presented twice in Portugal, at Ectopia Experimental Art Lab in Lisbon and I3S at the University of Porto, each involving a different version of the title artwork, When Microbes Dream. When Microbes Dream is an art installation that includes performative human and non-human interactions and hands-on biotechnological protocols. Together, it presents a feminist science fiction where biotechnology manifests interspecies collaboration, reproduction, theater, and storytelling as a means to reimagine our shared biotech future. The exhibition consists of 4 works. The first being the Biosafety Puppet Playhouse, which is a puppet theater that is also a biological safety cabinet. And it is decorated with this sort of Baroque Rococo patterns, but are also based on the local ecology here in Windsor, Ontario, Canada. The next piece is called Baroque Biology Paper Theater. This is a series of digital prints that are photographs of GMO bacteria in petri dish environments, laid and displayed on backgrounds of theater settings and designs. They're intended to create this image of the bacteria performing for us, sharing for us their information, their knowledge of biotech processes. Laboratory Ecologies is a complex installation that involves a sort of tent camping aesthetic for a lab that involves large animals, small animals, and microbes all working together in a cacophony towards a biotech research project. There are no humans in this installation. The humans are only the observers, the learners. And the final piece, called When Microbes Dream, the title piece for the exhibition, is a wall mural of microbial ecologies layered with a liquid spiral installation on top, giving this dream world, thinking about what it might be like for the microbes to live and cohabitate with other organisms in our lab. With this project and many others, I perform an alter ego character, the gentleman scientist, the epitome of the colonial traditional scientist who becomes absurd and defunct and self-deprecating as he or she moves through this biotechnological space, sharing with audiences access and discussion to biotech projects. Each artwork presents a complex vision of organisms large and small, mammals, microbes, plants, and insects, working in collaboration to communicate biotech processes to human audiences. This strategy deprioritizes the human scientist as the dominant organism in a robust laboratory ecology, rather presents the perspective of non-human organisms living, reproducing, performing, and dying. Of course, this is an absurdist and impossible task to ever imagine what biotechnology is like from the perspective of our microbial collaborators. It can only ever be a vision of what humans might think that microbes might dream. When Microbes Dream is produced with the support of the University of Windsor, the Incubator Art Lab team members, and the microbes and organisms that we share our lab with.
contaminated soils, communities saddled with lack of employment. These are the legacies left behind by the decline of the mining industry in South Wales. However, this contaminated soil can now be used for the production of something called a quantum dot, with the help from a very familiar garden friend. So, what exactly is a quantum dot? It is an incredibly small semiconductive particle. It emits different coloured light in different sizes when excited by light or electricity. They have been used in a range of different applications such as biological imaging, improved LED screens and solar cells. The industry is set to be worth over $11 billion by 2026. In 2013, researchers at King's College found that they can be biosynthesized using the earthworm through a process called mutual antagonism. When earthworms eat soils containing metals, they produce enzymes called metallothionein in its gut. The metallothionein grabs hold of the metals and stores it away in special cavities inside the cells. It reacts with another toxic element to make a safer compound. This is the quantum dot. Professor Mark Green of King's College explained,
"Extracting a quantum dot made in a worm is a cheaper, more sustainable process than making quantum dots in the laboratory."
Quantum Industries is one company striving to exploit this phenomenon. Working with a disused coal mine, they have biologically engineered a giant hybrid earthworm optimised for high-yield production of quantum dots. These earthworms are hatched in secure wormeries inside the mine. To attract the wormer runs a light electric current into the soil. The worm is then tickled to release the fluid containing the dots. This mine uses its cadmium and selenium contamination to create a cadmium selenide quantum dot. The BBC interviewed Margaret Howell, CEO of Quantum Industries.
Though we intend to sell quantum dots on as a green and cheaper alternative, our real aim is to generate clean energy using the quantum dots in our solar flags.
Quantum Industries are now fabricating innovative solar flags using spray-on solar cell technology. The flags are raised around the mine, powering itself and the nearby community. Quantum Industries hopes this would re-establish mines as a source of energy, bioremediating the coal industry's pollution and revitalising the industry in the area. Olivia Bargman, BBC News, Blenheim.
いつか出会い愛し合う二人は運命の赤い糸で結ばれている。そんな神話のような糸を紡ぐ蚕が2016年この世に誕生した。アートと科学の創造で新しい神話が生まれる時代始まったのかもしれない。
To me, there's so much awe and mystery in the way that life is formed. It's an intelligence beyond our understanding for all these parts to come together in perfect union. I created this as a piece of artwork, but in the future, we could potentially use this same method to make bone grafts that are implantable into the body. And if we're going to be implanting back into the body, what does that mean? How would we redesign our bodies, and what would we make ourselves into? As an artist and designer, it's also really interesting to consider what would we make if we no longer need to use inanimate objects like metal and fibers. If we can now use the building blocks of life, actual cells and tissue, what would we make?
This is Martha and George, who are soon to be mother and father. They made the choice to fulfill their pregnancy with parturients. Parturients, which provides artificial wombs for anyone for the period of 9 months. This is the parturients pod. The baby is nurtured through this object. It is similar to a pregnancy, but without the physical restrictions. Throughout this period, the family can live and work as they would normally do, whilst also being able to bond with their child. George A. Award, 29, is a journalist who is engaged with several interesting projects, due 2051. This is a communication device, which is connected with the parturients pod. The device can record desired moments of the daily life of the family. With its integrated microphone, it can record music or any other sounds desired. This is how the baby gets used to the voices of its future parents. Martha Aylward, 30, CEO of a company she started, works to ensure that the company meets their goals. It is almost time to go home. This is a portable communication device used when not at home. In between her meetings and lunch breaks, she can communicate with the baby. By simply switching it on, you can feel the baby kicking and moving. Carefully touching and rocking the device will send movements to the pod, which the baby can also feel. George is setting the table for dinner. There's Martha, home from work. George has chosen a wine to go with their dinner, and that's alright because Martha isn't carrying a child. Martha is cutting some ingredients for a special recipe that will set the preference of taste for their future child. The most important part of the growth process is the nutrition intake and the future preference of taste for the baby. The pod keeps track on what the baby has eaten and what it needs to stay healthy. After the food is prepared, it is placed in a capsule and placed in the parturients pod for the baby. At night, the pod goes through the updates made by the hospital with the information obtained during the day. This ensures the baby is healthy and the pod's program works without errors. The day is almost there. This is the final moment in which the pod's lock is lifted and the baby finally meets the family. George, Martha, and newly born Robin, one big happy family. Look how happy they are.
My name is Dr. Jennifer Willett. I'm an artist. I am a professor in the School of Creative Arts here at the University of Windsor. I'm a Canada Research Chair in Art, Science, and Ecology, and I'm the Director of Incubator Art Lab. I'm here to talk to you today about a new project called When Microbes Dream, an exhibition at Kapelica Gallery in Ljubljana, Slovenia in October 2021.
Since then, the exhibition has been presented twice in Portugal at Ectopia Experimental Art Lab in Lisbon and I3S at the University of Porto, each involving a different version of the title artwork, When Microbes Dream.
When Microbes Dream is an art installation that includes performative human and non-human interactions and hands-on biotechnological protocols. Together, it presents a feminist science fiction where biotechnology manifests interspecies collaboration, reproduction, theater, and storytelling as a means to reimagine our shared biotech future. The exhibition consists of 4 works. The first being the Biosafety Puppet Playhouse, which is a puppet theater that is also a biological safety cabinet. And it is decorated with this sort of Baroque Rococo patterns that are also based on the local ecology here in Windsor, Ontario, Canada. The next piece is called Baroque Biology Paper Theater. This is a series of digital prints that are photographs of GMO bacteria in petri dish environments, laid and displayed on backgrounds of theater settings and designs. They're intended to create this image of the bacteria performing for us, sharing for us their information, their knowledge of biotech processes. Laboratory Ecologies is a complex installation that involves a sort of tent camping aesthetic for a lab that involves large animals, small animals, and microbes all working together in a cacophony towards a biotech research project. There are no humans in this installation. The humans are only the observers, the learners. And the final piece, called When Microbes Dream, the title piece for the exhibition, is a wall mural of microbial ecologies layered with a liquid spiral installation on top, giving this dream world, thinking about what it might be like for the microbes to live and cohabitate with other organisms in our lab. With this project and many others, I perform an alter ego character, the gentleman scientist, the epitome of the colonial traditional scientist who becomes absurd and defunct and self-deprecating as he or she moves through this biotechnological space, sharing with audiences access and discussion to biotech projects. Each artwork presents a complex vision of organisms large and small, mammals, microbes, plants, and insects, working in collaboration to communicate biotech processes to human audiences. This strategy deprioritizes the human scientist as the dominant organism in a robust laboratory ecology, rather presents the perspective of non-human organisms living, reproducing, performing, and dying. Of course, this is an absurdist and impossible task to ever imagine what biotechnology is like from the perspective of our microbial collaborators. It can only ever be a vision of what humans might think that microbes might dream. When Microbes Dream is produced with the support of the University of Windsor, the Incubator Art Lab team members, and the microbes and organisms that we share our lab with.
Contaminated soils, communities saddled with lack of employment. These are the legacies left behind by the decline of the mining industry in South Wales. However, this contaminated soil can now be used for the production of something called a quantum dot, with the help from a very familiar garden friend. So, what exactly is a quantum dot? It is an incredibly small semiconductive particle. It emits different coloured light in different sizes when excited.
All right, everyone, welcome back. If you could please get settled, we're going to begin our afternoon programming, including a performance, which I'll introduce once everyone's settled, and the awards ceremony. So, yes, if you could please start getting settled in, that would be great. All right. Okay, everyone, welcome back to our final few hours of BDC Summit 2024. Can we get a quick round of applause for all the work and presentations that have been shown today? So I'd like to introduce our final performance of the day. It'll be a performance by Smita Sen, who is an artist working with sculpture, technology, and performance to study the relationship between the body and memory. Sen investigates haptic memory, the memory specific to touch stimuli, and considers how the body internalizes its environment in significant life events. With this approach, Sen studies how the human body can become a living document, storing tension, movement patterns, and emotion over the course of a lifetime. She will be presenting the piece Rituals of Sacred Surrender, developed by Sen and close collaborator and violinist Trina Basu. At its most beautiful and most turbulent, palliative care is an act of surrender for both the patient and the caregiver. Negotiating the competing qualities of the sacred bond, of the sacred surrender between caregiver and patient, artist Smita Sen will be delivering this performance to honor the complexities of palliative caregiving. So please give your attention to Smita's performance. Thank you.
Just need a moment. Thank you, Smita. I'm trying to calm down a little bit. That was a wonderful performance. I think during this time of the evening, afternoon, we're all kind of up here, right? And like there's a lot of anxiety, there's a lot of adrenaline, and I really appreciate, Smita, that you brought us kind of back to our cores. So, now we're going to the piece of the agenda that says special words. So here are the special words, I guess. I'm actually quite excited for this next part. Many of you know that over the last year, I've been working with the BDC board to find a new leader for our organization. We recognized that for BDC to continue growing and thriving, it needed someone who could bring new ideas and perspectives to our community. 6 months later, and after many interviews and many references that were called, we have found a wonderful fit. And I'd like to bring this person up to the stage to share just a few words. Before I do that, I would like to say, well, actually, let's bring Paul up to the stage. So everyone, please welcome to the stage Paul Rubery. I'm going to say just a quick. So Paul comes to us from the Suffolk County Vanderbilt Museum. He is an accomplished curator and nonprofit champion, and was actually the first to commission Laura Splan's guided sublimation performance, which we all saw this morning. Paul, I'm very excited to have you join us. I'm not reading this now. Thank you. It's been such a pleasure to get to know you over the last few months, and I'm really excited to have you take the helm.
Thank you.
I'll let you say a few words.
To be clear, you said 10 minutes. So thank you, Vina and Michael and everyone for the kind words of introduction. Thank you also to Dan, Orkon, Allison, BDC board members for inviting me to join the organization and to share some remarks today. I also wish to express my heartfelt gratitude to everyone seated in the audience, the instructors, students, parents, and BDC alumni who have attended Summit 2024. For 2 days, I have sat in a state of amazement learning about your projects and visions for the future. And for 2 days, I have been given an occasion to reflect on what it means to be a young biodesigner and how, thanks to your efforts, the world might become better despite the profound challenges that lay ahead. As Vina noted, I will soon step into the executive director role here at the Biodesign Challenge. Among other things, this means that we will be in communication, an active part of each other's professional communities as we progress toward common goals and interests. After hearing so many fascinating presentations, it feels selfish to direct attention to anything other than the wonderful work that you all performed over the past year. However, I would like to take this opportunity to briefly share some biographical details about myself so that we might come to know each other better. I joined the Biodesign Challenge after having served most recently as the Director of Curatorial Affairs at a regional natural history museum and planetarium on Long Island. Before that, I was a doctoral candidate in the Department of Art History, Theory, and Criticism at Stony Brook University. My intellectual commitments in both cases were located in the scientific humanities, specifically in physics and in the various zoological subdisciplines. My doctoral research concerned the ontological repercussions of quantum mechanics and the philosophy of photography. My museum work, the preservation and interpretation of historic taxidermy. Looking back on my professional life to this point, I have been privileged to study how the advancement of scientific knowledge provides us with analogies and methods for understanding human problems outside the domain of science. I think fondly on the books, conversations, and people who have shared in this concern. But most importantly, I recall the invigorating conferences, some very much like the BDC Summit, that compelled me to refine my worldview at a formative stage in my education. The transition I'm about to make into the world of biotechnology and design excites me because it extends that process. Biodesign indexes so many concepts that will provoke me to further evaluate my worldview. To shift now, as I am about to, from subatomic physics and its questionable materialities to a design-centric discipline with animate matter as its medium may, however, strike many of you in the audience as a counterintuitive step in the arc of one's career. After all, biodesign interpolates the biodesigner into a matrix of ethical quandaries, biological possibilities, and real human needs. It is unlike physics, or taxidermy, or anything else. Biodesign is transdisciplinarity at a different scale. But I do believe that my background in the history, philosophy, and sociology of early 20th century physics grants me unique perspective on what could be the discipline's future. It is this background that will inform the remainder of my comments. In his now famous formulation, the American biotechnologist Craig Ventnor once suggested that if the 20th century was the century of physics, the 21st will be the century of biology. I'm confident that some of the students in this room have heard that said before. In 2024, it would be premature to judge the validity of this claim, but there are undoubtedly affinities that lend it credence. If we humor Ventnor, and if we assume a one-to-one rate of progression, then in the biological disciplines we find ourselves, a quarter of a century later, neck and neck with Louis de Broglie's doctoral thesis on wave-particle duality. In other words, the biological century is in Act 1 and easing into the drama. Today's innovative biotech products, devices, and laboratory experiments are mere premonitions of the basic and applied work that will happen before 2099. That observation should give us hope and caution. Plenty can go awry in the next 75 years. But we should entertain the analogy because it is instructive for us here and now. The physicists of the early 20th century read widely, thought dangerously, and acknowledged the ethical complexities of the world they helped create. That world, the world of nuclear and particle physics, was unlike the one that preceded it. Not only was it shaped by the atomic bomb and other advanced weapons, but it was textured by the fallout of traditional Western metaphysics. Such extreme change in a short period of time still escapes description almost a century later, past and future disarticulated. The effect more devastating than the Copernican revolution. Simple, intuitive concepts like linear causality were now objects of doubt. Like all of us here today, the physicists and physics-adjacent dreamers of the last century stood at a precipice. They knew that their activities held tremendous consequences, and they felt the weight of that responsibility. In the face of true uncertainty, many of them made pronouncements which, from the perspective of today's critical theory, read like grand narratives, but in fact they were doing the work of ontologizing a world of their construction. J. Robert Oppenheimer, Niels Bohr, Richard Feynman, Albert Einstein, names that should be familiar to you, interrogated their labors and situated them in a broader social context. From them, we learned much about the ethics of uncertainty, the logical ordering of human experience, and the grammar of belief. Their commitment to thinking universally laid the groundwork for the most productive period of scientific description in human history, enabling physics to cannibalize all the sciences, or quite nearly all the sciences. 20th century physics confirmed that biology is special. Its central mysteries and concerns escaped the embrace of physical description. Erwin Schrödinger attempted to bravely define biological life in the 1940s, but ran against the paradox and its mysteries remained enshrouded. It is that enduring mystery that makes life significant and an object of reverence. It undergirds the responsibilities of the biodesigner. Historically, of course, life has meant many things to many different people. Approach an information theorist and you will encounter a definition markedly different from that of the generalist or the virologist, the surgeon or the priest. To the ancient Greeks, following the lead of Aristotle, life was a form of self-motion, perpetuation, or alteration. It was a reworking and a growth. As I step into this community, the Aristotelian understanding of life inspires me to meditate. I am particularly drawn to its principle of entelechy, or that quality in things which realizes or makes actual what is otherwise merely potential. Curiously, the concept of entelechy describes, for Aristotle, aspects of the relationship between form and matter, a hallmark of design. Does biodesign require a definition of life? Can you, the young biodesigners in the room, positively impact the world while leaving certain questions unanswered, select terms undefined? After seeing your fantastic projects, I certainly think so. What's more, I would like to believe that the Biodesign Challenge plays a significant role in realizing or making actual the potentialities of your impact, and I'm very grateful to be part of it. At the Biodesign Challenge, we are committed to 3 goals. First, to create a community of collaboration among artists, designers, and biologists. Second, to build meaningful public dialogue about biotech and its uses. And third, to seed the first generation of biodesigners. Our emphasis on futurity complements the profound resonance that exists between the Greek conception of life and the activities of our community. An orientation toward that which will be characterizes the promises of both enteleki and design. Yet mapping so many entanglements gets confusing. As we work toward a better future, we might struggle to know ourselves in a bramble of self-identity relations and near-perfect overlaps. Life, enteleki, design, design, enteleki, life. But for clarity, we can direct our attention to processes and actions. We could think of the principles for working and for crafting a better world. After observing you all this week and reflecting on the history of 20th century physics, I wish to offer some guiding maxims to the students in the room. Young biodesigners, together we must demonstrate humility in the face of ignorance. When facts are available and known, we must possess courage to state what is true. In engaging with others, we must be transparent because our activities affect the public and trust breaks easily. Anthropocentrism no longer makes sense, so throw it out. We must reevaluate our traditional conceptions of agency. We must be sensitive. We must know when to quiet ourselves and to listen to nature's teachings on ethics, growth, and self-realization. And we must make noise to root out injustice where we perceive it. Look always to learn from others as you progress in your education, whether it's at the BDC or beyond. And be comfortable reworking or abandoning beliefs you once held strongly. If you follow some of these rules, it will be easier for you to translate your most nebulous dreams into actualities. For the instructors in the room, more practical matters, please consider me a resource. My BDC email will be active shortly, and I encourage you to use it. Share your successes, trials, and thoughts with me. Tell me how the organization can further your educational and professional goals. If it is possible, I will certainly strive to help you. And for the parents and friends in the audience, and I did see some over the past 2 days, please know that the people that you are here to support are directing their cognitive resources, ingenuity, and passion toward the common good. Be proud of them, continue to show them your love and enthusiasm, and know that, among other things, they have the right stuff for living. Thank you all so much. I consider myself extraordinarily fortunate to have attended this year's summit and to have witnessed such promising minds at work. To the broader community, I look forward to discovering the ways we might enrich each other's lives. I'll see you next year.
Thanks, Paul. I was like, yeah. Okay, cool.
All right. Now the moment we've all been waiting for. No, we were waiting. Okay, so we're going to start with the Outstanding Digital Submission. This was a new prize that we introduced this year. For classrooms that aren't able to come to New York City, we want to still make Biodesign Challenge an accessible thing, right? We want the students to still be working alongside us, with us, our team, and to be able to share the work with their community and beyond. So we're going to introduce one of our outstanding display judges. Sorry, not display, oh my God. Outstanding Digital Submission judges up to the stage. Megan Valenitas, if you'd be willing to join us.
Sorry, I'm making sure the slides are up.
Thank you, Michael. Hello. It is my pleasure to be presenting the prize for Outstanding Digital Submission, as Vina said, on behalf of the virtual judging panel. This category had the added challenge of crafting a story not only explaining the team's innovation, and process, but to do so employing the craft of the moving image. The submissions in this category were far-reaching and provided a range of problem sets for our panel's consideration. The team we are presenting this prize to identified a relevant problem local to their community and performed extensive research both in the academic and design senses. While in our deliberation, our panel acknowledged the need for future material testing and the need to consider climate implications of their outcome, we were impressed by the breadth and depth of their iterative design process and problem finding. We could not deny this team's achievements across all evaluative criteria, exceeding in the categories of human and environmental impact, process, and narrative. We are very pleased to present the Outstanding Digital Submission Prize to Team Mirage from the Design Village. Congratulations.
We're going to quickly play their video for you all, just so you know who won. Thank you, Megan.
In Rajasthan's landscape, where colors reveal stories of wealth and tradition, there's a fading narrative, white. The starting hue of all has woven dreams in history, culture, and economy. In the deep crevice of towering marble slurry, a paradox unfolds. Where waste becomes beauty, people marvel at a majestic view, a magical paradise in Kishangarh among industrial remnants.
Exploring to find out when this started, we went to Kishangarh's busy street, learning about marble slurry and how it's made. The initial stage involves leveling the block using a blade, ensuring a seamless cut through the marble. Water running over the surface carries away the dust, marking the beginning of slurry formation. The block is then taken to be converted into slabs using the gang saw, where almost 20-30% of marble is transformed into the marble slurry. At this stage, constant water dripping is required for the blade to function smoothly as it cuts through the block.
Like a river, the slurry flows through the drains, encircling the plant. Starting with force at high pressure points, akin to a river's source, it gradually calms down, finding its way to the tanks below. The tanks are then transported to the marble slurry dump yard, where the slurry is deposited. Over time, the slurry dries out and begins to turn back to powder. The powder now swirls through the air, carried by sweeping winds everywhere. Yet as it spreads, a problem becomes clear. Breathing becomes a challenge for all, far and near.
Kids around the place enjoy playing in this very hazardous industrial waste, unaware of the fact that it causes health problems.
आप जब खेलते हो आपको कोई दिक्कत होती है?
नहीं।
मजा आते हैं?
मजा आता है पर पाउडर से गली जाता है।
आपका नाम क्या है?
मैं गुराम कुमार।
और आपकी उम्र कितनी है?
44 साल।
आप यहाँ कितने साल से काम कर रहे हो?
5 साल से।
अच्छा, और यहाँ पे मतलब आपका क्या काम रहता है यहाँ पे?
मेरा ऑपरेटरिंग का काम है।
और भी स्लरी से कोई दिक्कत होती है आप लोग की?
नहीं, हमारे कोई दिक्कत नहीं है, पानी के साथ चलती है, न उड़ती है, न कुछ होती है।
Cows graze in the marble slurry dump yard, suffering from numerous health issues. As designers, we imagined, what if we saw them not as a threat, but as resource treasures? We asked, what are they made of, and what can we create from them? Marble slurry is composed primarily of calcium oxide, along with silicon oxide, aluminium oxide, iron oxide, magnesium oxide, and moisture. Can we redefine this linear trail? From Kishangarh's depth, a fragment of marble slurry emerges. As it makes its way to our campus, does it carry the whispers of a large context challenges, waiting to be redefined and brought to full circle?
So we looked into what people were doing with marble slurry in our initial research, and we found 4 major directions that people were working on, and those were bioremediation, hydroxyapatite extraction, calcium solubilizing bacteria, 3D printing material, and composite material. So we chose to work with composite material because during our research we found that the composite material could be embedded right back into the micro context of Kishangarh where the marble slurry problem exists.
The journey continues with gelatin. By boiling gelatin with glycerin and water, an organic binding agent emerges, complementing the marble slurry and adding a touch of rigidity to the tiles. Initially, we experimented with binders like gelatin, which exhibited strength and rigidity upon drying. However, we encountered issues such as water solubility and animal collagen content. Subsequently, we dwelled into the natural binders like xanthan gum, cornstarch, potato starch, spirulina powder, agar-agar, and carboxymethyl cellulose. Following these explorations, we turned to the industry-use binders that offered more water resistance and strength to the marble slurry, such as terracotta, cement, grout, and resin blends. By mixing marble slurry with white cement in precise proportion, the slurry transforms into a solid, resilient tile, a testament to innovation and sustainability. Next, combining marble slurry with grout results in durable, sturdy tiles, perfect for withstanding the test of time and elements.
Building upon this, we conducted rigorous tests to assess material properties. Through these evaluations, we observed a variety of characteristics associated with the materials.
We tested the strength of carboxymethyl cellulose, potato starch, grout, gelatin, and cement. Both gelatin and cement demonstrated strength by withstanding hammering, while the others broke under the impact. After this, we tested the cooling properties of the tiles. They demonstrated significant water absorption and showed a temperature drop of 7 to 8 degrees after water was poured, making them noticeably cooler than before.
We proceeded with the exploration of cement binders due to their ability to resist water, easy availability, and low usage requirements, typically ranging from 10 to 20% in the composition. After testing all materials, we discovered that cement exhibited strength.
We found that due to the high porosity of marble slurry, it remained cool for a long duration of time. This brought us on the path to experimenting with tiles and cladding made out of marble slurry that would act as a passive cooling mechanism. On that trajectory, we looked at various forms that would enhance this property as well as provide additional structure to maintain the longevity and porosity of the tiles. Taking inspiration from passive cooling forms, Jali in Rajasthan emerged as a functional solution which provides both sunlight and ventilation. Jali uses the Venturi effect in which the hot air from a high pressure region moves to an area of low pressure and turns cool.
We began by experimenting with various 3D forms. After sketching our final design, a triangular hyperbola, we rendered it in 3D software. We created an inverted triangular hyperbolic shape to reduce air pressure, ultimately harnessing the Venturi effect. Next, we used computational fluid dynamics to test our material. We calculated its density and porosity. In the following screens, you will first see the pressure drop by 5 bars as air passes through. We also simulated the material in Rajasthan's weather. Without water, the temperature drops by 3 to 4 degrees, and with water, it decreases by 10 to 12 degrees.
In our journey of marble production, we have come full circle crafting tiles from marble slurry that preserve its essence and cool touch.
However, the challenge lies in the affordability of marble slurry, priced less than 1 cent. While easy to obtain, transportation cost often surpasses the material's value. It's imperative to address the marble slurry issue locally in Kishangarh. If it is left unattended, it will continue to accumulate in the dump yards, posing risks to water quality and public health. By harnessing marble slurry, we not only overcome these challenges but also create employment opportunities for the community. Establishing a manufacturing unit in Kishangarh will complete the cycle, turning industrial waste into a valuable resource. During our ethnographic field research, we spoke with mine and dumpyard workers. Off camera, they acknowledged the problem, but on camera, their statements changed, and suddenly, marble slurry was no longer an issue. This denial is pervasive. Our project aims to enhance the well-being and work life of these workers, addressing the challenges they face. Everyone knows the issue, yet their eyes remain closed. Isn't it the time to face the problem? Isn't it the time to open our eyes?
All right, everyone. Thanks for watching that video, and thank you to the Design Village for their thoughtful submission.
It's 1:30 in the morning back home there, so I hope y'all are there online. Congratulations.
So the next prize that we're going to announce is Outstanding Instructor. And so to present this prize, we'd like to invite a past BDC instructor and now BDC judge, Betsy Wissinger, to the stage.
That's okay. So this is, okay. I have the great pleasure to announce that the outstanding instructor is Devin Ward of Auburn University.
And as Devin's coming up, just to give a little bit of context, we always ask students for nominations for their professors. And the letters that were written for him were moving, to say the least. So congratulations, Devin. Up next, we're going to do our special prize. I hope he's here. We talked a little bit about the Science Sandbox Prize yesterday. To announce it, we have Omar Vera, our friend from Simons Foundation. Omar, are you here? Okay, there he is. Yeah.
Hi, everyone. It's so great to be here with you all in this space. I just want to start by saying thank you to Veena and Michael and the whole Biodesign Challenge team for this amazing event that's happening here. My name is Omar Vera. I'm the program specialist for the Science, Society, and Culture Department of the Simons Foundation. Within that sits Science Sandbox, which is our flagship grant-making initiative, as well as the namesake for this prize. We believe that everyone should have a relationship with science that is authentic to them and to their lived experience. Partners in events like Biodesign Challenge help bring that to light by introducing new and creative ways to interact with science that affirms, celebrates, and centers a person's identity. This is ultimately the goal of the Science Sandbox Prize of Public Engagement, to think about how we can meet a community where they are and make sure that everyone has a voice in the way that our science and our technology advances, and develop new futures for ourselves and for our communities. I want to thank both specialists for their participation and for thinking so critically and creatively about what public engagement means for their project. Without further ado, I'm happy to present the winner of this year's Science Sandbox Prize for Public Engagement, EASD Castello.
Okay, so for the next prize, we're going to be announcing the Outstanding Art Prize. And this is an exciting new collaboration we're doing with Biobat Art Space. So we'd like to invite Elena Sotorakis up here to describe this new collaboration and announce the prize winner.
Hello, everybody. I'm super thrilled to be here today and partnering with the Biodesign Challenge, and thank you to Vina and Michael and all of the inspiring presentations today. So for this award, the Outstanding Art Award, I'm the co-founder and curator at Biobat Art Space, which is a bio-art gallery located in the Brooklyn Army Terminal in a biotech incubator. And so we're dedicated to interdisciplinary collaboration and advancing bioart. And for this prize, we're proud to announce that we will be mentoring this group for a full year. We will be, in June 2025, we will be giving them a solo show, and we will also be giving them an honorarium, an artist stipend to advance their project. So many of you, I know, we're working on very limited time constraints. And so for this team, we were really looking at a group that was working very well together collaboratively, that could do a lot with more time, space, and financing. So I'm pleased to announce that the winner of the Outstanding Art Award goes to pHen from Moholy-Nagy.
I'm loving the compliments as we're taking the photos too, thank you. Gas us up, gas us up. All right. Up next, outstanding field research. We are gonna bring up another collaborator of ours from the Cambridge University Press, Martin Dade Robertson. Why do I keep saying Robin? I'm sorry. And Giovanna Danis. Yeah.
Well, that went well. You can tell we haven't rehearsed this. So thank you so much for inviting us to give this prize today. So we've been running a journal, Biotechnology Design, with Cambridge University Press for about 2 years now. And honestly, the inspiration for the journal was this community and our experience of being here. So it's reinforced that, I think, for both of us. Why are we doing this? The journal is about inviting early stage research, interdisciplinary research that wouldn't usually find a place in an academic journal elsewhere. And we've seen so many examples this week of stuff I really want to publish. So If you see us after this session, please do find us, and we'd be more than happy to talk to you about publications and so on. Whether you're in the final 8, whether you've won this prize, or indeed whether you've been here for the last 2 days, we're interested in talking to you. So,
and the winner is
CCS Salt Mines.
All right. So for this next prize, we're going to be announcing the Outstanding Display. It's a new prize this year because we noticed that so many of you were putting in so much work into your gallery displays, and it never held a part in judging or had a prize. So we really wanted to celebrate and honor those of you who put effort into that element of the summit. So to invite one of our judges who will be presenting the award, he was actually a secret judge, for those of you who didn't know, going around like talking to y'all, like trying to figure out like who had the best display. And so I'd like to invite Akash Marai up to the stage.
Thank you. So as Michael has said, this prize is about celebrating the wonderful displays that you have all presented yesterday, which allows us to engage with and discover your journey through your projects and learn about what all the story was and what was happening there. The winner is one of the displays which the project was aiming to engage with the community, and the display was as engaging as the community they were engaging with, and allowed us to get hands-on with the materials and play with it. It was a project which turned charcoal into ink from the ashes from Auburn University.
Just moving right along. Okay, cool. Up next, we have Outstanding Narrative. To present this award, we actually are working with Twist Bioscience this year to support the winner of this award. And to present it, we have Dante Lombardi.
Thank you. This award is all about storytelling, and it occurs to me that stories can be the most powerful force for good in the world. Stories and storytelling can be the most powerful force for evil in the world. The right story can persuade you to change your life and improve your life and change the lives of the people around you. The wrong story can persuade you to destroy your life and destroy everything around you. Now, it's unfortunate, but a new kind of science has developed, a new kind of computer science designed to deliver as many of those wrong stories to us as possible every single day. They're with us all the time. And I think the last 2 days, for me, and I think for all the other judges, have provided an important antidote to that. Because we heard stories of science, we heard stories of design, and we heard stories of hope. And that's tremendously inspiring, and it felt authentic. And the team that we are going to award this prize to had all the elements. Their story had great context, great characters. It had a narrative arc. It had drama. It had great science. It had great design. It had great humanity. And some of the words that I heard in their presentation that really jumped out at me were things like care, collaboration, mend, regeneration, self-repair, resiliency, lungwort lichen, and quilting. And so that's why I'm proud today to award this narrative award to MICA and Symbiotic Stitches.
Your turn?
I think so.
Okay, go.
All right, so up next, we've got Outstanding Science. And we've seen so many projects that could embody outstanding science today, and so we wanted to invite someone from the community who's also really fostering outstanding science, and that is Leila Tavares from GenSpace, who will be presenting the award for this Outstanding Science Prize.
Hi everyone, I'm Leila. I'm the education manager at GenSpace. If you don't know about GenSpace, we are the world's first community biolab, a place where we have people from all backgrounds come and be able to get into the sciences. So if you want to learn more about us, please catch me after we end here. And we're super excited to be able to offer this prize this year with the BDC. The people who, or the team that will win this prize, they actually will be able to have access to our community and resources. So we're really excited to be able to support them, whether that is through continuing the project that they presented here or maybe developing new ideas. So the winner of the Outstanding Science Prize goes to Shall We Dance from California College of the Arts.
With the grand prize. It's not up there. Okay. Up next, we have Outstanding Social Critique. So for this prize, we are working with New Inc., which is the incubator space for the new museum. Biodesign Challenges, actually, we were members of their program in 2019, and we have stuck with them since. You can't get rid of us, and we are anchor tenants there now. So the winners that will actually, I'll let our judges talk about it. So up to the stage, I'd like to invite Anye and Raul. Sitting together, ready to go. Love it.
everybody. Let's give a round of applause for the Biodesign Challenge team. Vina, Michael, Cody, somewhere out there. You're rocking it. Star alumni at New Inc. My name is Raul Bengetch. I'm the Deputy Director of New Inc. Like Vina said, we're a museum-based incubator for people with art, design, and technology practices. We run a one-year program where we help people level up their creative projects. I like to think that there will be more than one of you in the audience who will join our program soon, but in the case of this award, the winner will gain a community membership at New Inc., which is an on-ramp into our program for future years. So to introduce our Social Critique Prize, I'm going to say a few things of what that means for us. Critique is an act of love and care. Transformation is something that happens from the inside. Radical change, as Adrienne Marie Brown describes, from the root. We protest, we refuse because we care deeply, and we honor the possible worlds we create when we practice critique. We at New Inc., as an institution, are nothing without the artists, designers, and researchers whose work keeps us working. Today we award the prize to one project, but tomorrow it will be the person sitting next to you. Remember the ones you come up with. Liberation is a forever process, a state of mind. It is undying.
And in this world where there's so much greenwashing all around us, and there's a system of innovation that caters to economic growth rather than real change, I think this award is more important than ever, I think, at this time. And who we're going to award this to starts to ask the question of what happens in a system where economics and growth is centered rather than real change.
This is for everyone. Never stop fighting, never stop loving, never stop pushing us to be the best versions of ourselves.
And with that, we'll announce that the winner, after a really tough deliberation, is Urban Vein from NTU.
We're going to keep the applause going until everybody comes up. Woo!
And like another one of those like cool community moments, we had posted about mentorship at New Inc. on our Instagram, and Anye signed up for it, and now he's one of the New Inc. mentors as well, which is really fun. Just a fun story. Okay, good.
Okay, so next up we have Outstanding High School Project. So this is a new prize we introduced this year as well, because we always have high school teams that participate and always stun us with their projects and thoughtfulness, despite their point in life and the experiences they've had. And they use that as their strength in their projects. And so we really wanted to reward and award that. And so to do so, we wanted to invite one of our past BDC high school instructors who's always championed that to present this prize, and that'll be Corinne Takara. So if you could please come to the stage.
I don't know about you, but some of the most inspiring projects I see are from the high school students. So I just want to give a round of applause for all the high school students that participated. It takes so much courage to come here and compete against adults. And it's wonderful that this year we actually have a category for these students, and I hope they will continue to participate and be competing again from universities in the future. So it's my honor to uplift and celebrate Relaxance Grace Church.
Okay, I'm nervous again. Why am I nervous?
Because we're announcing the last two prizes.
Oh. Okay, last two prizes. Up next, we have our runner-up prize. So I say this every year, and I'm going to say it again. We appreciate all of the hard work that each of you puts into your classes when you get here, when you present. These prizes are just one piece of the larger piece of BDC, right? So whoever wins, whoever doesn't, we will always, Biodesign Challenge will always support you. We have evidence of so many years of students who, like, whether they won or not, they would go on to do wonderful things, right? So just know that this is a great prize and this is a great thing to experience to get this, but there are more things that will happen after the summit. So please do stay in touch with the Biodesign Challenge. We love to work with you guys going forward. Okay, that being said, we'd like to invite our new executive director, Paul Rubery, to come and give the runner-up prize.
Should we set up the micro?
No, I like to pull. It's fun to pull it out of the case.
Where is it?
This one.
All right, runner-up, Eunice Conbay.
It just came so fast. I was like, oh, that's great. That's great. Okay. Oh God, we gotta do all the pomp and circumstance of getting it out of the Pelican case. All right.
What could it be? Oh, it's a glass microbe. Wow, that's the grand prize. Who's gonna be introducing this one?
All right. Wait, can you hold this so I can put the case away and don't drop it? Can you imagine?
It's a lot lighter than I thought.
Right? It's so light. Okay. I'd like to introduce Vina and Michael up to the stage to announce the...
Oh, I'm here.
Michael! Okay. All right. Do you want to say something?
Sure.
Okay.
So I guess maybe for me as program manager, it's been for the first time. I was just a lowly program associate last year. But in this role, it's been amazing to work with you all, see the growth of all of your projects. I know most of you all only knew me via email. And so when you were coming up to me like, oh, you're Michael. You send me all the emails. I'm like, yeah, wow, I didn't know it meant so much to you.
You're like a celebrity. We can stop you on the streets.
Yeah, it's been amazing seeing the culmination of all your work, and especially seeing you here in person, and just getting to talk and hang out with some of you at the student mixer. Like, that was a great experience. Like, just having these touch points to make these connections. It's been really cool seeing some of you all meet each other, you know, create and cultivate these connections.
Hey,
the theme of the gallery show. Wow, here in person. And so I think seeing those things happen, those subjective, less quantifiable experiences happen here at the summit, has been ultimately the most rewarding for me. Absolutely. And so this prize, you know, goes out to all of you, but specifically, it goes out to the actual winner.
Whoa! We're in the end. Never mind. I'll stop. Okay.
And I think this winner embodies the spirit of BDC. Their project was so thoughtful, so fun, dipped into the science, dipped into the art, dipped into the design, dipped into the critique, and also just really showed that passion that we're trying to foster here at BDC.
And really made us respond to, just to speak to the judges' room, there was a lot of discussion, there was a lot of debate, so I think you did what you set out to do, so thank you for that.
Yeah, and so without further ado, I'm going to announce the winner of the grand prize of BDC Summit 2024.
You gotta hold it up, not awkwardly, don't hold it up awkwardly.
I don't know how to not do it awkwardly. All right, the winner is UT Austin Consumer Consumption.
That was so much fun. Okay. Okay, we got just a few more things left. Again, congratulations to everybody. Can we do? Our team is very much excited to work with you all in the coming year, whether you were a winner or not, right? Whether you were in the top 8 or not. I think we were inspired by so many of your projects. There are only so many prizes that we can give out, right? But there were so many of you that were so close. That's not a fun thing to say, I'm sorry. But like, to say that it was a really hard decision on every single prize, because so you all stepped up. Like, our team, multiple times we're talking about how this year, you were so impressive by how much research you did in the work in your projects, how well you answered judges' questions. Like, many of you were anticipating it, and you knew what needed to happen next. You were very reflective in the work that you were doing. So yeah, we're proud of all of you. Thank you. It's okay, we don't have to clap again. It's gonna. Okay, so we're just gonna do a few final thank yous to close the night out. Firstly, again, thank you to Science Sandbox, the sponsor of the Prize for Public Engagement. We appreciate you all. Thank you to the Siegel Family Endowment, New York City Cultural Affairs, the National Endowment for the Arts, New York State Council on the Arts, Cliff Family Foundation, BASF Foundation, Twist Bioscience, and Tom Tex. Thank you again to all of our collaborators that we mentioned on day one, Grow.bio, Amino Labs, New Inc., Cambridge University Press, LifeSci NYC, BioBat, and GenSpace. Thank you to every single one of our volunteers. We had a force this year. We had a workforce that was incredible, and we were so happy to have you all with us. We couldn't have made the day happen without you. So thank you for being reliable, showing up on time, and just being wonderful people. Thank you to the MoMA team, Sean, Carson, everyone up there. Can we just give the AV department just... behind the scene people always have my heart, because I know that life. Thank you to our BDC board, Dan, Allison, and Orken. You guys are amazing. And finally, can I bring our staff up to the stage, please? So Cody, Afifa, Bhakti, Alex. Alex, I know you're here. Where are you? There she is. She's coming. Okay, cool.
Shout out to Cody. Cody was our event producer, event coordinator, made like all of this happen, no joke. So shout out to Cody. And then, of course, also shout out to our wonderful interns, Afifa and Bhakti. It's been so fun working with y'all over the course of the past few months, and you know, answering all of your Slack messages when you're very confused about what we're supposed to be doing. And you always pull through, so thank you all. Thanks, Dina. And of course, Alex. It's been so nice working with you again for this summit time, and I'm so glad to have you back in this capacity, so thanks, Alex.
Michael just did the work for me, and I think this was like a great microcosm of the entire last year. So I'd like to, again, thank you to all of you. I think there were moments where each of you just like, I was losing my shit, and like you stood up and you like backed me up, right? Like, and throughout this entire last year of me just trying to figure out leadership and working with our board and lots of different things, I felt very supported by all of you. So thank you, thank you, thank you. But a special, wait, we're not done yet. But I, like, above all else, thank you to Michael for being just like my rock through everything, right? Like, I think, I'm gonna cry, so I'm not gonna say anymore. But I, like, just, yes. I appreciate you, and I'm,
Yeah, I just wanted to say it's definitely bittersweet, you know, that this is your last year. You know, the past year working with you, coming off of last summit, Dan leaving, now you leaving. It's like, the past year has been so much fun, and I've learned so much from you, and you've been an amazing mentor and friend.
don't do it. If you do it, I'll do it.
I got it under control, don't worry.
Okay.
But yeah, thank you for everything.
Okay, cool. Now that we can stop gushing on stage, we appreciate all of you. Thank you again for this wonderful year. We're so excited to see what happens next year, and go forth and enjoy New York City. Oh, wait, no, hold on. We had two people. We had Elena come up, talk about BioBat, and Layla came up and talked about GenSpace. We are doing excursions tomorrow, so if you all are still in New York City for the weekend, I know many of you have already signed up, but if you have not, come up to us and we'll talk to you about it. You can sign up to go see, there's gonna be a curatorial tour at BioBat led by Elena, and then there's gonna be open labs, so you can go and check out what GenSpace is all about. So, yeah. If you're still here, join us for that. And thank you. Congratulations to everybody. Wonderful job. See you in 2025. All right. Oh, I forgot to tell the wait. Fuck. Hello. Oh, also, I'm so sorry. Students that have prizes, students that have won prizes, we're going to take photos of you in the garden. Just stay here, and we will take you to the garden to take photos. And we will also give you cases for your prizes so you don't drop them.